Whenever you guys want me to start, I can.
So we're very close to the total of registrations. If maybe we just want to start in a minute. I think
Okay,
we're good.
let me give the intro.
83 right now. Somebody's asking if this is going to be recorded.
Yes, I am replying to the chat right now. So, but yes, the answer is it is being recorded.
Okay. Give me one more second here. Video's not picking up. Okay, everybody, thank you all for joining us today. Can you hear me? Am I live?
Yep.
Good. All right. So it's CE training time again. This is our 9th consecutive year. I want to start off by giving a little plug for Napa Farm Bureau, the organization that I put these meetings on through. We're a grassroots organization with the sole goal of protecting agriculture in Napa Valley, and it's an important cause. I think it affects every one of us probably in one way or another if we're on this call, and your ongoing support is the key to future success. Big thank yous to Hugo Navarez, who is instrumental in putting this thing together. He is the nuts and bolts and glue and all of it, and wouldn't be possible without all his hard work. Corinne and Kristen as well at the Farm Bureau, and the Napa Ag Commissioner's Office. They've been partnering with us for 9 years on this, and helping us achieve the laws and regs component that everybody needs to keep their licensing. Over the course of the trainings, we have 4 trainings. If you're not signed up for all 4, please consider signing up for all 4. You can gain 20 CE credits total, 16 other and 4 hours of laws. And so if you come to these meetings every year, you've got your CE requirements covered. I'm really excited about the subjects that we're going to be touching on this year. I strive to keep these things timely and interesting and new, and I think we've succeeded with that again. And the beat goes on. So without further ado, I'm going to introduce our first speaker, Dr. Cristina Lascano, with the Department of Land, Air, and Water Resources at University of California, Davis. Dr. Lascano is a soil biologist and assistant professor of soil ecology. She completed her PhD at the Soil Ecology Research Group of the University of Vigo, Spain. Afterwards, she worked as a postdoctoral scholar at the Technical University of Denmark, the University of Calgary, and UC Davis. Before joining the faculty at UC Davis in 2019, she was an assistant professor of soil ecology at Cal Poly, San Luis Obispo. And the basis of her research at the most basic level is to identify soil ecology interactions responsible for nutrient availability and the retention and nutrient retention in soils. I'm sort of reading, excuse me for stumbling over that, and how they can be managed to increase sustainability in ag production settings. I will stop there. The title of Dr. Lascano's presentation today is Members of the Soil Microbiome and Their Contributions to Healthy Crops. Thank you for joining us, Christina.
Thank you very much, Eric and Hugo, for your invitation and Napa County Farm Bureau. I'm really honored to be here and excited to be talking about my favorite topic, which is soil biology. So I'm a soil scientist, but my bachelor's degree is in biology. So most of my teaching and my research focuses on the living components of soils and how they are responsible for crop productivity and also sustainability. Um, so you probably heard a lot about soils, but mostly from the physical or chemical aspects of soils. And that's what we've known pretty much for the last decades or even 100 years. But we don't know so much about the living component of soils. Who's living there and what are they doing and why does it matter? However, for the last maybe 10, 20 years, the advancement of molecular techniques has allowed us to understand much better the tremendous amount of biodiversity that lives in the soil. And so now we know that soils are amongst the most diverse ecosystems on Earth, and virtually all life kingdoms are represented in soils. So we have from eukaryotes like fungi, plants, and animals to prokaryotes like bacteria and archaea, and even viruses. And in addition to that, we have organisms of all sizes. So you can see here in this picture, microorganisms like fungi, bacteria, archaea, viruses, all the way to larger organisms like earthworms, moles, and everything in between, such as, for example, nematodes, and you have here an example, mites, you have another example here, Collembola, etc. So, and in addition to this, all of these members of the soil biome are extremely abundant. And just to give you an example, in the case of microorganisms, we know that for a very small amount of soil, like a teaspoon of soil, which is maybe like a cubic centimeter, like a few grams of soil, there are more microorganisms that people are on Earth. And so that's really mind-blowing, right? But what's most important is not just who is there, the identity of the biota, or how many of them are there, but how they interact among themselves and also with their physical and chemical environment. That type of interactions can be many times summarized or explained through what we call trophic interactions. These trophic interactions describe how organisms feed on each other, and they can be represented for what we call food webs. And so here in this slide, you have an example of what it's a simplified soil food web, which is showing the different trophic levels, starting with the first one, which is the primary producers, the plants and the organic matter. The second one are the decomposers, so the microbes that decompose organic matter, but also the plant feeders. So there you have your nematodes, your plant parasites that feed directly on the plants, right? Then on the higher trophic level, we have microbial grazers, and shredders, and some predators. And higher trophic levels, like the fourth and the fifth here, have other types of predators, which are also larger in size. As an example here, you have moles or spiders, right? So the most important thing is that by interacting and feeding on each other, this food web and the biota in this food web regulate the transfer of energy and matter through the whole soil, right? So the energy that of the sun that is captured by the plants is transferred all the way to larger organisms through this whole food web, all right? So these interactions are extremely important. Interactions of soil biota regulate very important soil processes, such as the cycling of nutrient, decomposition of organic matter, plant growth and photosynthesis, and even the formation of soil. And these processes are essential for supplying the soil ecosystem services, such as the provision of food, fuel, and fiber, the provision of genetic resources and pharmaceuticals, the sequestration of carbon and regulation of the climate, the composition of waste, purification of water, disease and pest regulation, conserving biodiversity, etc. All right. It is the interactions of the soil biota that are responsible for the provision of these ecosystems. And I want to emphasize the fact that soil biota supports the two major life-forming ecological processes in nature. On one hand, soil biota supports decomposition, which involves the breakdown of biomass to release nutrients. And on the other hand, soil biota supports net primary productivity by releasing those nutrients and allowing the plants to capture them. In this process, as plants grow, plants sequester CO2 from the atmosphere. They capture it into the biomass. Through the process of decomposition, part of that carbon is going to be sequestered in soil. It's going to be stabilized in soil together with nitrogen. At the same time that this is happening, there might be some release of greenhouse gases from the soil, such as CO2, nitrous oxide, and methane. All right, so the same process of the composition involves a certain degree of greenhouse gas emissions. This is a natural process, but what we need to understand is that soil biota, by regulating the composition and net primary productivity, is regulating atmospheric composition. And this is something really important that we need to keep in mind when we manage soils. And you probably heard about the potential of soils for carbon sequestration and climate change mitigation already. This is something that has been in the media for a long time. And several organizations are actually trying to promote this type of climate change mitigation strategies. I want to highlight first that this is not a solution. This is just part of the solution. If we want to address climate change, we obviously need to reduce emissions first. But managing soils and promoting carbon sequestration can help draw down part of the CO2 that is already in the atmosphere. And so to give you an idea, soil organic matter has 58% carbon. So increasing soil organic matter has a large potential to increase soil carbon and remove it from the atmosphere. To give you an idea of the significance of this practice, a 0.1% increase in carbon per acre is equivalent to the emissions of an average car per year. So if we think about Napa County having 45,000 acres planted on wine grapes, that was in 2017, increasing, if all the growers were to increase their soil carbon by 0.1% per acre, that would mean removing the same amount of cars from the streets for 1 year. So this is really striking, right? The potential of this type of practices. And not only soils can help mitigate climate change, but they can help also adapt to climate change. We know that with climate change, there is more likely for droughts to happen, and water scarcity here in California is a big problem. And we also know that increasing soil organic matter can improve the physical properties of soil, like reducing bulk density, promoting aggregation, and higher porosity in the soils. This, on one hand, improves infiltration and water percolation, but on the other hand, also improves water holding capacity of the soils. So this is a number that has been put together by the USDA and RCS, and they say that as an estimate, for every 1% increase in soil organic matter can result in up to 20,000 gallons of available soil water per acre. So I want to again emphasize that this is an estimate, and this is a number that is being highly contested right now. So this is a potential increase. It doesn't mean that it's going to happen every time you increase soil organic matter, right? There's a lot of variables that will affect this. But in fact, we know that soil organic matter improves water holding capacity. So this increased understanding of the importance of soils for crop production, but also environmental quality and climate change mitigation has spurred worldwide efforts to support soil health. What is soil health? Soil health has been defined by also the NRCS, the Natural Resource Conservation Service of the USDA, as the continued capacity of soils to function as a vital living ecosystem that sustains plants, animals, and humans. So here you have in this definition, the biota, the living component of soil as that vital living ecosystem, which is fundamental for supporting soil health. So our healthy soil is characterized by supplying a series of services and functions and having a series of properties that are associated not just to crop productivity, but also with the physical, chemical, and biological properties of soils. So a healthy soil is supposed to not only support high yield and high crop quality, but also whole diverse soil organisms that are active. And by being active, the soil biome or the soil microorganisms actively sequester carbon, they reduce the emissions of greenhouse gases, and they cycle nutrients, but at the same time, the cycling of nutrients is tight enough so to reduce losses of nutrients to the environment. For example, reducing nitrate leaching and greenhouse gas emissions. Finally, a healthy soil is also characterized by physical properties that make it store water and have a good water infiltration and drainage. So I want to emphasize as well that the soil properties of a healthy soil to be targeted through management or to be looked for are way differently for different crops, because not all crops have the same needs. And also depending on the production goals and site-specific challenges, certain soils have for example, higher challenges with physical properties. So we might want to focus more on fixing the physical properties of that soil, or some others have very low contents of organic matter and nutrients. So we might want to emphasize the increase in soil carbon and nutrients, right? Focusing on crops, not all crops are the same, not all crops have the same needs. So that means that the meaning of soil health may be different for different contexts as well. So for example, the meaning of soil health may be different for crops like corn or wine grapes. In fact, most of the soil health indicators and management guidelines have been designed for crops which are different than wine grapes, mostly crops from Midwestern regions of the US, such as corn and soybeans. And on the other hand, we know very little about wine grapes and what is the definition of a healthy soil for growing wine grapes. So for corn, for example, we know that corn has high nitrogen requirements. So what we want of a soil is that it's able to support high yields. However, in wine grapes, the priorities might be different. Maybe we want high yields, but we also for sure want to have a certain crop quality. We focus very much on a certain chemical profile on the grapes, right? On corn, on one hand, what we want to support to that high yield is a soil that supplies high amounts of nutrients, but also retains the nutrients. We don't want to have losses through leaching, right? In the case of wine grapes, having a lot of nutrients is actually not good because it promotes vegetative growth, right? So we want a soil that retains and recycles nutrients, but doesn't have a lot of nutrients available. And we also want to focus on a soil that stores water because of the particular context-specific challenges that we have in California, right? And the droughts. So there are lots of open questions currently in relation to soil health and wine grape production. And this is what I want to focus on the rest of the presentation, and I want to show you some of the research I'm working on dealing with these topics. First of all, we need a definition of soil health in wine grape production. We need to define what are the most important soil properties that produce high quality grapes. And so what are the soil aspects that we need to management, that we need to target through management? And also, we need to understand the importance of certain novel soil health indicators like the importance of the microbiome or soil carbon in supporting soil health and wine grape production. Once we understand that, then we need to tailor management practices so we can enhance those soil properties for high quality grapes. So I'm going to start with the first question, which is the definition of soil health for wine grape production. The importance of soils for wine grape production is nothing new, and the wine industry is probably one of the most aware of the importance of soils for the production of quality grapes, right? The impact of soils on wine quality when combined with climate, topography, and grapevine variety, it's what we know as the terroir effect. And there are several soil properties, mostly physical and chemical properties, that are well known to have an effect on grape quality and vine yields, right? And this chart is showing some of the examples of the properties that have been the most researched so far that are considered to be components, the soil components of the terroir. Those properties are physical properties of soils, like the depth of the soil, the texture, the structure of the soil, the infiltration, but also chemical properties of soil, such as the cation exchange capacity, the mineralogy, the pH, the nitrogen supply, all right? We know quite a lot about these properties and how they are related to the production of wine grapes. Now, the definition of soil health, if you remember, is slightly different to what we know of the terroir. So some of the properties and soil health indicators that are included in the terroir definition are also included in the definition of soil health. For example, soil structure, infiltration, water holding capacity, and nitrogen supply. But the concept of soil health brings new indicators to the table, such as soil organic carbon, microbial activity and biomass, biodiversity, aggregate stability, etc., that haven't been considered before as components of the terroir, okay? So the question is like, shall we start thinking about these variables as also important components of the terroir? We propose that yes, because they might have a very important, very remarkable effects on the vines and on the composition of the grapes and the quality of the wine. If you want to know more about this topic, how the definition of terroir and soil health overlap, I recommend you to go and check out our article that was just published in Frontiers in Sustainability. And it's free for download. So you search for terroir and soil health and Lazcano, and you should just find it and you can just download it for free. So overall, we need to start considering new components of the terroir. And in particular, we need a better understanding of the role of soil biota and the soil microbiome in driving the production of wine grapes and the quality of wine grapes and the effects of that in the quality of the wines. The soil microbiome drives important processes such as nutrient cycling, the control of diseases, carbon sequestration that might impact the quality of the grapes. but also microorganisms from the soil we know that are transferred into the grapes either through the plant or directly through dust. And those microorganisms are transferred into the fermentation process. So microorganisms from the soil, either directly or indirectly, will affect the grape quality and the quality of the wine at the end. And so many researchers are starting to talk about a new concept, which is a microbial terroir, all right, which defines how the microbiome affects the quality and chemical profiles of the grapes and the wine. These are questions we have started to work on recently. We have a project which is looking at the variability in soil health indicators into the context of Napa Valley terroirs. This is a project that is carried out by the PhD student at UC Davis, Noelimar Gonzalez, and also in collaboration with Kerry Steenworth from USDA, Toby O'Gene, Malika Noko, and Khan Kurteral from University of California, Davis, and with support from the American Vineyard Foundation and in collaboration with the Napa Valley grape growers. So the objective of this project is to provide a better definition of soil health for wine grape production, and also a baseline of soil health indicators in the region of Napa Valley. So to do that, we're carrying out, on one hand, semi-structured interviews with growers and vineyard managers to put together a definition of soil health based on the knowledge of the growers. And on the other hand, we're doing an extensive landscape soil sampling to establish soil health indicators for the region of Napa. We want to specifically address different types of soils in the region of Napa because we know that there's a high diversity in soil types, which actually shown before to be able to sequester different amounts of carbon. And so potentially they might have absolutely different levels of soil health and potentials to increase soil health. This is a study that was done by Kerry Steenworth and Kyla Burns in 2015. And this is a map of Napa County here with all the different appellations. And so here you see for those different appellations, the total carbon that you can find in those soils. And as you see, some of them have low contents of carbon, but some of them can have really high contents of carbon. So there's the type of soil and the location matters a lot. So we really need to understand this, how the type of soil is driving soil health. And so this will help growers understand how much organic matter increase is reasonable to expect for a specific soil, which has a specific texture. We know that clay soils, for example, accumulate more organic matter than sandy soils, okay? So how much increase in organic matter are we to expect for a given soil? How much microbial activity, how much nitrogen, etc., right? So establishing this baseline will help us also create realistic expectations on what to expect when we are managing to increase soil health. A really important component of this project also is focusing on the microbiome. So we want to understand what are the most important drivers of the soil microbiome across all of the different soils in Napa Valley, and how is the microbiome related to soil carbon and soil health. To do that in the same soils that we're going to collect, we're going to do analysis of DNA markers using 16A RNA for bacteria and ITS markers for fungi. And we're going to do analysis of PLFA biomarkers to assess the living soil microbial biomass and fungal to bacterial ratios in the different soils. This is something that we're going to do in collaboration with the biotech company Biomarkers, who have agreed to fund the molecular analysis of our soil samples. So we're really excited about the potential outcomes of this project. For now, this is all what I can say. We don't have any results so far. This is a project that started this year. We just got funded this year, so we are collecting samples right now, but we hope that in a year, we'll have, a year from now, we'll have more data to show. We're really excited about this project. All right, moving on. I hope everybody's still doing okay. I want to transition to the second of our questions, which is, once we understand the role of soils in supporting soil health and the role of the microbiome, and we have a better definition of soil health for wine grape production, the second question is, how can we manage the soil biome to support wine grape production and reduce the environmental impacts of this production? So in terms of management strategies, management for soil health is based on three main principles. First of all, the increase in soil organic matter. So practices such as organic fertilizers like compost and manure are really good for increasing soil organic matter, but also using cover crops or incorporating crop residues. The second principle is to reduce disturbance to the soil by, for example, keeping the soil covered, using cover crops to avoid runoff and erosion, and reducing till or using no-till. And the third principle is the increasing diversity within the operation, such as using crop rotations. This is obviously for annual crops, but using cover crops in the case of perennial crops like vines will help increase diversity within the vineyard. All right, so those are the basic principles of healthy soil management: increasing soil organic matter, reduction on disturbance, and increasing diversity. There has been a lot of research over the last 10 to 20 years that showed that practices such as compost application, no-till or reduced till, and the use of cover crops are really good at increasing soil organic matter and increasing soil health. However, the outcomes of these practices are extremely variable. So for the same practice, which is being applied in different soils or different regions, we can have absolutely different results. Compost application might work in one vineyard and not work so good in another one, all right? And not only that, but in addition to this variability, we still lack a good understanding on how the changes in soil health may affect the quality of the grapes. The reason for that is that because we're missing a piece of the puzzle, which is the effects of the practices on soil biota. And when we understand this better, we can manage for specific soil health outcomes and specific grape chemical profiles, okay? So we currently work in different projects studying the effects of compost application, no-till, and cover crops on soil biota, soil health, and grape quality. But for the purpose of this presentation, in the next slides, I'm going to be focusing just on compost application and organic fertilizers, all right? The first experiment or project I want to talk about, it's the project of Holly Denniston-Sheets. She was a master's student at Cal Poly working under my supervision, and she studied the effects of organic fertilizers applied to vineyard soils on the soil food web. This was a project in collaboration between researchers from Cal Poly in San Luis Obispo and University of California, Davis, and was supported by the Agricultural Research Institute of the California State University, and was carried out at the Paragon Vineyard of Niven Family Wine Estates. The objective of this research was to assess the effect of fertilizer inputs, which included organic but also inorganic fertilizers, on soil health by using nematode food webs as proxies. Why nematodes? Well, if you look at this simplified soil food web here, you will realize that nematodes are really well represented in many of the different levels of the food web. We have nematodes that feed directly on the plants. Those are the plant parasites or herbivores. We have nematodes that feed on microorganisms. Those are microbivores or bacterivores or fungivores. Okay, there's nematodes that feed on bacteria or on fungi. And then we have nematodes that are predators. So they feed on other nematodes. Isn't that cool? And then we got also nematodes that are omnivores. So they eat whatever, microorganisms, other nematodes, plants, etc. So there is way more nematodes than your typical plant parasites, right? And nematodes are actually extremely important because of all of these different roles in the food web in supporting nutrient cycling and decomposition. So studying these nematodes, it's a good indicator of the health of the soil food web. All right. So nematodes are very good indicators of soil health, not just because they are represented in all of the different trophic levels, but also they are sensitive to disturbance, and so they change with soil management. They're well correlated with beneficial functions like the cycling of nutrients and carbon sequestration, and they're very useful for elucidating ecosystem processes. So in soil ecology, we love nematodes. In this field experiment, we actually compared not just fertilizers, but also cover crops and different types of strategies for weed management. So it was a very large project with 3 sub-experiments. But as I said, I'm just going to focus on the fertilizer experiment, which you can see here. This is an aerial view of the vineyard. And so this trial here in the middle, these plots, was our fertilizer trial. Okay, this vineyard was in San Luis Obispo County. It was a Pinot Noir vineyard in Etna Valley. And fertilizers were applied to these blocks under the vine for 2 years. And the organic fertilizer was a mix of poultry manure and feather meal. Synthetic fertilizer was an NPK fertilizer, which was a standard for vineyards. Within the plots, we wanted to look at 2 different locations, the soil under the vine and the tractor row, as you can see here in this picture, because they are fundamentally different in the management that they receive. The soil under the vine, first of all, receives treatment to remove weeds. In this case, it was mechanical weeding, but receives also the fertilizer and water through irrigation. The soil in the tractor row does not receive fertilizer, does not receive water, anything else than precipitation. And there's usually a cover crop. So there's active vegetation growing there for almost like the whole wet season, right? So when we looked at the nematodes in the 2 locations, we saw really different abundances, okay? This chart is showing the number of nematodes per cubic centimeter of soil in the soil in the tractor row and the soil under the vine. So there were way more nematodes in the tractor row, so in this middle row, than under the vine. The abundance of nematodes was different in the 2 locations. And not only that, but the types of nematodes that we found at the 2 locations was also different. And this is what I'm showing here in this chart. Again, the bar chart here is showing just the abundance of nematodes. This pie chart is showing the different types of nematodes. I mentioned before that we got herbivores, we got fungivores, bacterivores, predators, and omnivores. All of the different trophic groups within nematodes. Herbivores here are marked with orange color, fungivores with yellow, bacterivores with green. So as you can see here, the vine row, which had lower number of nematodes overall, were also mostly dominated by herbivores. So those are your bad guys. Those are the ones you don't want to have, right? On the other hand, the tractor row, the amount of herbivores was much lower, and there was a better balance between the good guys, the bacterivores and the fungivores. Those are the ones that consume microorganisms and promote the cycling of nutrients. So the diversity and abundance of nematodes was higher in the tractor row. And that was also associated with other soil health indicators such as soil pH, available nitrogen and carbon that were also higher in the tractor row, most likely because of the presence of the cover crop. Now, when we looked at the soil under the vine and how the fertilizers applied the nematodes, we seen that the fertilizer had a significant effect on the type of nematodes that were found under the vine. They were actually able to ameliorate the nematode structure under the vine. So organic fertilizer increased, you can see here in this chart, increase the abundance of fungiborne nematodes as compared to the synthetic fertilizer and the no fertilizer, which was our control. Also, the organic fertilizer, you can see here in this bar chart here, the organic fertilizer increased as well the abundance of bacterial nematodes under the vine. So applying organic fertilizers increase the abundance of those beneficial nematodes. So it ameliorated the abundance of plant parasites under the vine and increased the complexity of the food web. In this experiment, we looked also at the quality of the grapes and the yield of the vines, and we've seen that for most of the chemical parameters that we looked at in the grapes, like Brix, pH, total acidity, berry weight, and berry set, the fertilizer treatments did not have an effect on the quality of the grapes. However, we've seen a significant effect on the yields, on the kilograms of grapes produced per vine. What we've seen is a reduction, a significant reduction in yields with the organic fertilizer. We still don't know why this is happening, but it's remarkable. So we didn't see any effects on the quality of the grapes, but we see an effect on the yield of the grapes. Okay, so for the last part of the presentation, this is the last experiment I want to talk about. In this experiment, we studied the effects of compost application on soil carbon and greenhouse gas emissions, also in wine grape production. This is the master thesis project of Connie Wong. It's a master student under my supervision at Cal Poly. And it's done in collaboration with other researchers at Cal Poly as well, such as Jean Dodson-Peterson, Charlotte DeCock as well. So the objective of this study was to determine how applying different rates of compost may maximize soil carbon sequestration while reducing environmental impacts. We wanted to find what's the best rate of compost application. So we have the maximum amount of carbon sequestered in soils and with the minimum production of greenhouse gas emissions. And remember that increasing soil carbon may actually be offset by the releasing of greenhouse gases like CO2 and nitrous oxide and methane, which are naturally produced through the process of decomposition. Adding compost to the soil increases the amount of soil organic matter, increases decomposition rates. So it might actually also increase the release of CO2, nitrous oxide, and methane. And this is something that needs to be investigated so to reduce these potential environmental impacts of healthy soil practices. This was what we tried to do in this experiment. We're still working on this. This slide is showing you the design of this experiment. This was what we call a randomized complete block design. We had 4 rates of compost, 0, 2, 4, and 6 tons of compost that were applied to the vineyard, broadcasted to the vineyard. This is an aerial, what it would be an aerial view of how the treatments are distributed in the field here. We got 4 blocks, and within each block, we got the 4 treatments or 4 rates. Blue is 0 tons of compost per acre, green is 2 tons per acre, yellow is 4, and orange is 6 tons per acre. The type of compost applied was organic compost produced at cow poly. It's a mixture of livestock manure and green waste. The CN ratio is 10, around 10, so it changes from year to year. We applied this compost in 2018 and again 2019, and we are just applying it now for 2020. So it usually happens after harvest and before the first rain in fall. And again, the same as in the previous project, because of the really important differences in soil management between the tractor row and the vine row, we sample from the two locations. So we compare carbon sequestration and greenhouse gas emissions in the two locations, the soil in the tractor row and the soil in the vine. We collected samples after every compost application, not just of soil, but also greenhouse gas emissions by using static chambers. Here you have, this picture is an example of how we collect the gas samples in the field. And we also collected grape samples at harvest to analyze grape chemistry. I will show you the results now. And I'm gonna start with active carbon or POXY. Active carbon is a fraction of carbon that is actually very sensitive to management, and it's available for microorganisms to break down. So it's very likely to be stabilized. It's a good indicator of potential carbon sequestration as compared to total carbon in the soil, right? So this is something that we often look at in this type of studies, active carbon. In this chart here, you have the different rates of compost represented with the different colors, as I explained before, blue, green, yellow, and orange for the increasing application rates. And you have the amount of active carbon in milligrams per kilogram of soil for the soil collected at different depths, from 0 to 6 inches, from 6 to 12 inches depth, and from 12 to 24 inches depth, so pretty low in the soil profile. In the tractor row and in the vine row, the two locations, right? So what you can see here, you notice probably, is like a striking decrease in soil carbon with depth. This is very typical. Soil carbon is typically more abundant in the top soil than in deeper parts of the soil. The first 6 inches is where most of the carbon is. So that's what we saw here. But in addition to that, there was a remarkable difference between the carbon in the tractor row and the carbon in the vine row. Tractor row have way more carbon than the vine row, all right, in that top layer of soil. Other than that, we didn't see any differences of compost application rates on POXY, on soil carbon, okay? I'm sorry. We did see an effect of compost application rate on active carbon, and higher rates, 6 tons per acre, had significantly more active carbon than lower rates like the control and the 2 tons per acre. So applying higher rates of carbon may lead to higher rates of compost will lead to higher rates of active carbon in the soil. The next couple slides are showing the cumulative greenhouse gas emissions, nitrous oxide and CO2. I'm gonna start by explaining nitrous oxide. In this case, these charts are showing you the cumulative emissions, so the total emissions produced in the first post-treatment wet season. So that was 2018 winter, 2019 spring, which corresponds with dormancy. The first post-treatment dry season and the second post-treatment wet season. We're showing the gases split into dry and wet seasons because soil moisture and precipitation had a really strong effect on greenhouse gas emissions, and so the processes happening in the soil are different. So in this case, starting with the first wet season, we didn't see any significant differences of the treatments, either on the tractor row or the vine row, in the nitrous oxide emissions. In the first dry season, that was the first growing season after compost application, we did see a higher amount of emissions in the vine row than in the tractor row. And this is probably due to the fact that vine rows during the growing season, they are irrigated. So as I mentioned before, higher moisture leads to higher emissions. Soil moisture affects nitrous oxide emissions, and that might be the reason why the vine row had higher N2O emissions. The second post-raining wet season, which is 2019 winter to 2020 in spring, saw an increase in emissions in general, but no differences between tractor rows or vine rows or even different rates. I should also highlight that in any of these cases, we did not observe differences between compost application rates. So applying compost did not have an effect on nitrous oxide emissions, besides what we just saw of the effects of irrigation under the vine. In the case of CO2 emissions, again, we are showing the different periods, the two wet seasons and one dry season. What we've seen is that there was a seasonal pattern here. During the wet seasons, the tractor rows had higher emissions of CO2 than the vine rows. This happened in the first wet period and in the second wet period. However, this relationship was reversed during the dry season, where we saw higher emissions in the vine row as compared to the tractor row. This trend is probably due to the fact that tractor rows, as I mentioned, they have a cover crop growing. So during the wet season, the cover crop is green, is actively growing, and also there's higher root respiration, which leads to higher CO2 emissions from the tractor row. During the dry season, the cover crop is terminated, so the emissions for the cover crop go down, but the irrigation started in the vine row. And so that irrigation and higher moisture promotes higher microbial activity under the vine, and also that releases higher CO2. So the main takeaway messages from this experiment is that compost application can actually increase the amount of active carbon, which is observed up to 0.6 meters depth in the soil. So that shows a potential for compost to sequester soil carbon. In the case of greenhouse gas emissions, compost application and the different rates didn't have an effect on greenhouse gas emissions, either nitrous oxide or CO2. So that suggests that there are no environmental trade-offs in terms of greenhouse gas emissions with increasing application rates. And then what was the most remarkable of this experiment were the differences between the tractor and the vine rows. And this is because of the presence of the cover crop. So we think that actually the cover crop applied together with the compost might actually be boosting carbon sequestration, and there might be a synergy between those two types of healthy soil management practices. So applying compost and a cover crop together might actually increase the probability of sequestering carbon in soils. So to wrap it up, this is my last slide. What we learned from these experiments and these projects is that targeted management of the soil biome can help increase soil carbon and soil health in wine grape production, which has clear agronomic and environmental benefits. However, there's still a lot we need to know. And more precisely, we need to understand how important is soil biota as compared to physical and chemical properties of soils for wine grape production. We need to understand as well how much can we build soil health for different soil types, so we can have realistic guidelines for the different types of soils. And then, very importantly, we need to have a good idea of what are the actual effects of the soil microbiome as affected by compost or other management strategies on the quality of the fruits or the grapes, right? This is an idea we're working on right now. We just submitted a concept proposal to the CDFA, and we are hopefully getting funding soon. So we will be working on that over the next years. Thank you very much for your attention. I am really happy to take any questions. I think we still have a few minutes. I think we got, yeah, maybe like an hour allocated for this. But Eric and Hugo, you can let me know if we still have time.
Yeah, we've got time for a couple of questions. And Christine, if you wouldn't mind hanging out for a few minutes on the chat, any questions that we miss verbally, we can address, through the chat function.
Yeah, I see a question here by Molly. It said, like, it looked like there was higher N2O emissions in the no compost treatment. Was that insignificant? Yeah, that was insignificant. So there was a lot of variability between treatments. That's something that happens a lot with the type of studies, with this type of studies and with nitrous oxide specifically. But so in this case, it was not significantly different from the compost treatments.
Dr. Lascano, there is a previous question as well. I don't know if you're able to see it on the Q&A, but it says from Aaron, it says, when and how is soil organic matter measured? It is highly variable depending on soil moisture.
Yes, it is. Yeah, so we normally sample organic matter. It depends on the project, right? But we assess soil organic matter as total carbon in the soil, and that's usually analyzed by loss on ignition or combustion of the soil, and we measure the carbon that is released after burning a soil sample. And that's, as I mentioned, organic matter has 60% of carbon, so it's a really easy conversion to estimate organic matter from soil organic carbon. And it's a really stable, soil organic carbon is very stable. It takes some time to build, so it doesn't really change very quick. Within a year, it's not going to change a lot. Total soil organic carbon, I'm talking about. Then within total soil organic carbon, there are different fractions, like that active carbon I just mentioned. Those fractions may change more within the same year. And they might be more variable with moisture, for example. But total carbon does not change within a year, for example. It takes a while for total carbon to change. And then in terms of when we sample, most of the times we get samples in spring and like around bud break or maybe a little bit later. And then, but it depends on the project. For the compost application, sometimes we just take samples right after compost application, which happens now in November. I see that there is another question. Do you advocate the application of manure to vineyards? It's tricky because manure has a lot of nitrogen available as compared to compost. Compost has a lower amount of plant available nitrogen. And having a lot of plant available nitrogen for vineyards can be a problem. So I would be careful with that. What I recommend, it's a careful management in general, understanding how much nitrogen you're putting with whatever amendment that you're using and how that nitrogen is going to be released for your crop, etc. So understanding a little bit of the nitrogen cycling dynamics. I think compost is better than manure for vineyards, just because of that, for the available nitrogen. Do you advocate the application of grape stems to vineyards? Yes, and this is another study that we did. I'm talking about this actually. If you're attending Sustainable Ag Expo, which started this week, we have a presentation on compost application to vineyards. So I recommend you that you watch it, and we talk about this project, but also other projects going on in the state. And we talk about how grape pomace and stems and in general waste from winemaking, it's a really good source of carbon and nitrogen for soils in vineyards. So they should be reused, but they need to be treated before. They need to be composted. You cannot apply, obviously, grape pomace, which is fresh to the soil. It's bad. It's phytotoxic and it will pretty much kill everything. But if you do composting, and a lot of vineyard managers, they compost their own waste in on farm, then that's really good. It's a really good source of nutrients and carbon for your vineyard. So Stan Servas, did your greenhouse gas emissions calculations include the increased tractor time to spread 6 tons? No. So no, we did not include the emissions related to tractor operations in this study. That would be really interesting, but it was not the focus of the study. In this study we just focused on the soil, but it would be super interesting to compare those trade-offs, if you have to apply more compost and how that's increasing the tractor related emissions.
Okay, thank you very much, Dr. Lascano. In order to stay on schedule, perhaps we can take the rest of the questions offline. I want to say, again, thank you so much. That was fantastic. A lot of really good new information on a part of the vineyard that I forget to think about sometimes, and other people may as well. So again, thank you, and Ugo will be launching a poll that everyone needs to answer in order to qualify for the CE credits. Everyone's probably been through one of these before, but if you haven't, Ugo can kind of help to navigate that.
So I just launched 2 questions as a poll. They're multiple choice, and it's basically like a voting kind of thing. So you'll see the 2 questions regarding this first topic, and you would, for both of them, you can only choose one answer. And I'll leave it on for a couple minutes so everybody can just cast their answer, and at the end we'll export all the information and send it to DPR. One thing, Eric, that I did want to say really quick, earlier on the presentation, Charles asked about the article that Dr. Lascano mentioned during the first half of her presentation. I don't know if you could quickly remind everyone on how, I think you said how they could Google the article, or if you can share the link with us and maybe we can send it to everyone. And
I'm doing that right now. I'll put it on the chat.
Perfect.
Okay, while we're in process here, I want to say a big thank you to Sunridge Nurseries, our sponsor today. Julian Clymer is your rep here in Napa County, and if anyone's joining us from anywhere else, there too. So please contact Julian and have your grapevine plant material needs met. He's a board member on the Farm Bureau and very active with the young farmers and ranchers, and so really good to support him and his organization.
I just sent the link for the article. So as I mentioned, it's free to download, and yeah. Thank you very much.
Okay, thank you. All right, everybody, continuing with our theme of vineyard soils, our next speaker is joining us from Florida. He may be in the midst of Tropical Storm Eta, or Eta, however it's pronounced, as we speak. I hope it's passed over and it's not something that you need to worry about anymore, Johan. But thank you very much for being here. Dr. Destiger is a professor of entomology and nematology at the University of Florida. I'm going to give you his background here briefly. He grew up in Belgium and he's been working in nematology for 25 years. He started his career in Kenya and has continued to move around the globe, conducting research internationally for the industry, private industry, and university alike. He's been based in Florida since 2005 at the University of Florida's Gulf Coast Research Center, and he's focused on nematode management in fruits and vegetables. Thank you very much for being here, Dr. Desiger.
Thank you very much, Eric, for the introduction. And yeah, luckily our tropical storm, which turned out to be a hurricane, has passed. But we got about 10 inches of rain just yesterday. So a lot of flooding in the area. I heard our research farms got some damage to some of the hoop houses we have. But overall, we got lucky. But, you know, it's every year these storms start coming later and later in the year. So it's part of climate change, so that's something we're gonna have to deal with in the future. Let me, can you all see my presentation here, my slides?
Yep.
And I'm loud and clear?
Yes.
All right, sounds good. I wanna congratulate Christina also. That was a great talk, very interesting information, and very good to hear you're doing some work on nematodes as well. Maybe at some point we need to talk about that. So I'm going to talk about fumigant alternatives for nematode control. As Eric said, I'm based at the University of Florida, and I'm located here at the Gulf Coast, which is one of the 13 research and education centers of the University of Florida. We're here in Waimama, which is a small blue-collar town, farming community. Little bit like half hour south of Tampa. Because of the proximity to Tampa, the place is changing quickly. We're getting a lot of new developments being built. So, you know, there's a lot of pressure on the farming land, and I'm sure 5-10 years from now, that place is going to look very different than what it's looking at right now. We got a pretty nice research center there, about 200 staff, 40 students, 18 faculty. Our focus there is really high value crops. Do a lot of vegetable research, small fruits, strawberries, blueberries and such, and also some ornamentals. Put some pictures of my lab there, which is changing all the time. So we have some students, postdocs, biological scientists, and some technical support staff. So this is the outline of my talk. I'm going to talk first a little bit about nematodes, the unseen enemy. Talk about fumigation a little bit, some of the new nematicides that have come available recently, and I'll finish up with talking about biological management, and then hopefully there's some time at the end left for questions. So nematodes are great parasites. You know, every living organism on the planet is parasitized by some type of nematodes. In a lot of tropical regions, you can see the top left, that's a disease called elephantiasis. It's caused by a nematode, transmitted by a mosquito. Below that, you see a piece of salmon fillet, and in the middle, you can see this little nematode crawling in there. So fish has a lot of nematodes. Really everything we eat, every animal we eat can be parasitized by nematodes. And it's no different for all the plants, the crops that we eat. Annual crop loss is estimated at over $100 billion, but it's really difficult to estimate that because crop loss due to nematodes, the symptoms are really not very typical. You know, you may see some stunted poor growth, some slow decline with trees, some yellowing like you see in this grape here in the top middle. So it's often confused with other issues, whether it's disease or you know, other constraints. It's very likely that this will increase in the future, the crop loss. Nematodes are likely to get worse. There's about 4,000 species in 125 genera, but really only about a dozen genera cause the bulk of the damage across the world. It is considered one of the most serious threats to global agriculture in the future. So this is a typical plant parasitic nematode. They basically feed by piercing and sucking, kind of like you have with aphids and some other insects. You see this structure here at the top, which is what we call a stylet. It kind of works like a hypodermic needle. So the nematode uses it to puncture root cells and extract nutrients from it. Fairly complex organism, you know, they have a digestive system, reproductive system, several glands, small nervous system, even they're mostly microscopic, about a 50th of an inch long, about 1,000th of an inch wide. So they're very thin. That's why they're often called threadworms or eelworms even. The pictures here on the right top, you see a root knot juvenile penetrating a hair root, just to give you an idea of the dimensions here. And below it is a root knot female nematode, which becomes more round, pear-shaped on top of a needle pin. Just to give you an idea of the size. Okay, seems to be stuck here. Okay, here we go. You know, Christina was already talking about it earlier, but the bulk of the nematodes that live in our soils are beneficial, are not parasitic. So we call them beneficial or free living nematodes. And most of them feed on bacteria or fungi. And then there's a smaller number, these are usually larger sized nematodes that are omnivorous or even predatory, so can feed on other nematodes. And I'll talk a little bit about that later when I talk about biological management. This is just, I hope this video is gonna work. This is a sample of nematodes underneath a microscope. This is 40 times magnified. So this is a whole bunch of different nematodes that we extracted from soil. So we're going to 100 times magnification now. And this one here in the middle is a plant parasite. This is a Pratelenchus, a lesion nematode, and you can see the stylet here at the head. That is how we identify them. The one on the right here, this is a bacterial feeding nematode. There's no stylet here. They just have basically a mouth canal which they ingest bacteria. That's how they feed themselves. So this is typically the way they move. You know, they have this kind of slow, gentle movement, kind of like a snake. They're aquatic. So nematodes basically swim. They don't really crawl. They're swimming organisms. So they need water. In a dry soil, you will have hardly any nematode activity. So plant parasitic nematodes can feed on roots in many different ways. There's some that feed on above ground parts, on leaves and seeds in some cases, but the majority are root parasites. So that's what we'll focus on. But they can feed inside or outside of roots. So the most damaging nematodes, like your root knots and your cyst nematodes, they will actually penetrate the root. And once they're in the root, they become sedentary, so they don't move anymore. And their entire life cycle happens inside the root. So they develop these specialized feeding cells called giant cells, and that causes the typical swellings, the root swellings. Other nematodes like lesion nematodes, which you'll have in your vineyards too, they will actually enter the roots, but they'll keep their worm shape. So they stay mobile and they can move in and out of the roots. Most of your nematodes in the soil are what we call ectoparasites. So they feed on the outside, they just use their stylet to insert it into the root, mostly the root tips, and feed like that. So sting nematode is like that, stubby root, ring nematodes, your dagger nematodes, which you have in vineyards as well. They're all ectoparasitic nematodes. This is, you know, if there's one nematode that, or one plant parasitic nematode that you should remember, it should be root knot nematodes or Meloidogyne. This is the most damaging nematode in the whole world, certainly in Florida, but also in California. Causes these characteristic galls, swellings on the roots. It has a very wide host range. There's very few crops that are not affected by this nematode. The size of the galls can vary a lot depending on the species of root knot you have, as well as the crop that it affects. The one on the left here with the big galls is tomato root. Right next to it, this is hops, which is also a good host. See a lot of smaller galls, but huge, enormous numbers of it. Next to it is a strawberry, which has typically smaller galls on the roots. On the right here is another tomato. Bottom left here, that's an artichoke, which has really big galls as well. Tubers can have galls like yam roots, potatoes. So this is at least one nematode. When you dig up a root system, it can tell you whether you have a nematode problem or not. Most other nematodes, you will have to do actual soil and root extraction, so a lot more labor intensive. They are considered, this nematode, root knot nematodes, is considered among the most rapidly spreading of all pests and pathogens in the world. This is one that we have in Florida. You may not really have in California. It's really more restricted to Florida and the Southeast, really prefer sandy soils, but this is our #1 pest in strawberries in Florida, causing, really severe stunting of strawberries. This is an ectoparasite. It's a large nematode. It's actually can be several millimeters long. And it just causes this severe stubby looking root system. So it chews on the root tips, or it feeds on the root tips, and the root tips stop growing. And then you get these stubby looking roots, which really become very ineffective as far as water and nutrient uptake. That's why you get these severely stunted plants. As far as grapes in California, root knot nematodes are probably the major problem, and you have different species in California as well as we do in Florida. Dagger nematodes may be the next one. There's 2 species. One of the main problems with dagger, they can cause root damage in itself, but they are one of the few nematodes that can actually transmit viruses, causing yellow vein and fan leaf disease on grapes. And that's really the main issue. The picture here on the right shows you, this is from Mike McHenry, retired University of California nematologist, showing root knot damage on grapes. Here below you see the root system of a grapevine, typically relatively small galls. If you look at the red dots here, those are the egg masses that the females secrete, the female root knot nematodes. So these egg masses can contain several hundreds of eggs, and they're fairly easy to stain, to visualize, as you can see here. Some other nematodes in vineyards are needle nematodes, citrus nematode, root lesion is fairly common. Also ring nematodes in some places can be quite damaging, and there's also some sheet nematodes. I put some links here. There's some interesting, valuable fact sheets from University of California describing management guidelines for grape nematodes, so check it out. And I also put a Zoom link here, which is it's a recorded webinar just a few weeks ago that one of my students helped organize, focusing on nematodes in perennial crops. And there's a talk from our citrus nematologist at Florida on citrus, but there's also a talk from a nematologist from USDA in Oregon, Inga Zasada, who's done a lot of work on small fruits in Oregon and Washington, including vineyards. So if you want to know more about this, check out this webinar and this website. So fumigation. Why do growers fumigate? Well, the goal is really to reduce nematode population densities and allow for a successful plant establishment, which is, especially critical for perennial crops. For decades, really since the 1950s, this product here, methyl bromide, was the standard fumigant, especially in Florida and California. Growers really were dependent on methyl bromide for their soil fumigation. Of course, in the 90s, it became known that methyl bromide broke down the ozone layer, was one of the main products that caused it. So the phase out of methyl bromide started, and growers had to look for alternatives. So this was really a big deal, not just California, but certainly also in Florida, and especially for strawberry growers, which were completely dependent on methyl bromide at that point. So right now, there's a number of alternative fumigants that have replaced methyl bromide. The main ones are telone, 1,3-D, different kinds of metam-based products, and chloropicrin. Paladin here was out there for a while. It's dimethyl disulfide, but was taken off the market, I think, last year. So we have these 3 products. That's basically it. They are effective, but they're just not as broad spectrum as methyl bromide. So what growers have to end up doing is combine these products to get a good broad spectrum. Telone is your best nematicide. Chloropicrin is probably your best fungicide. And then metam products, you know, are pretty good herbicides, but are probably have more of a broad spectrum activity. So especially in Florida, fumigation is still the standard for nematode management in high value crops. So most vegetable strawberry fields are still fumigated. And this shows you the effect that a fumigant can have in a Florida tomato field. So fumigated on the left, non-fumigated on the right, and you do see a big impact of fumigants, which is why growers still really rely on fumigants for their nematode and soil disease management, if you will. Now, even fumigated fields, we can still see severe damage, and probably, you know, with the loss of metal bromide, disease issues and nematode issues have become more important for sure. This just shows you a few examples. On the left here is a tomato field, which was fumigated with C35, which is telone and chloropicrin, but you still can get pretty severe nematode damage. You see all the wilting and yellowing here, which was correlated with this quite severe root gall damage on these plants. Often in tomato fields, we see interactions between root knot and diseases like Fusarium wilt, which is what was going on here, and that really can, you know, exacerbate the damage to the crop. Watermelon here in the middle, again, this field was fumigated with C35, but still, we got a lot of dieback of watermelon vines here. The grower thought it was disease related. When we came by, we looked at the roots. It was pretty obvious that this was a nematode issue. And then here on the right, there's a caladium field in Florida, fumigated with not just C35, but he also broadcasted metal bromide. Caladium growers can still do that. And metam as a crop destruct on the previous crop. So we had 3 different fumigants here, and we still can get this, you know, pretty severe damage. So nematode, especially root knot nematode in Florida, is a really serious and difficult to manage constraint. So one of the key questions for nematode management when you see these failures of fumigants is one of the key questions here becomes, where are the nematodes? Nematodes do move. They're not like fungal spores or weeds that just sit there. No, they actually move. So this is some data from Joe Noling, a just retired nematologist at the University of Florida, who did some deep core sampling. He's got this piece of equipment that can go 3-4 feet deep. It's called a probinator. And it can take soil samples, you know, through the hard pan up to, typically we go about 3 feet deep. Technically we can go 4 feet. But this field here was sampled just before it was supposed to be fumigated in June. This is a central ridge soil, which is a very deep sand. And if you look at the map of Florida here on the right, this is this yellow area is where the deep sand is. So here, there was no crop in the field. And not surprisingly, we don't find any nematodes in the topsoil, but we do find significant numbers, you know, 2 feet and even up to 3 feet deep of root knot nematode. So if you just fumigate this topsoil, you're not gonna touch the nematodes that are hiding deeper here. And that's part of the problem. This is a different soil. This is now this green area here, which is what we call our coastal flatwood soils. They're more shallow, they're not as deep, they have a higher water table. They're still very sandy. I mean, all the soils here, green or yellow, are more than 90% sand. Now this crop, this field was planted when it had a crop. So it had an eggplant in this place, a greenhouse tomato in this place. When you sample during the crop is when you're gonna find most of your root knot nematodes in the top 1 foot. This is where most of the roots are. So this is where you're gonna find most of the nematodes at that point. But you still find significant numbers, you know, 1 to 2, even up to 3 feet deep. On the right here with a clay subsoil, so we only saw them to 2 feet, but you still find nematodes at depth at that point. So especially in the sandy soils of Florida, this is a big problem as far as, you know, nematode management goes, especially if you know that these nematodes, when they're here at the subsoil, they can actually move upwards quite rapidly. Some work from the early 90s here from Mochaidi showed that nematodes can migrate upwards up to 22 inches in the sandy loam soil. This was done in Oregon in only 9 days. So it doesn't take them much time. It is different in a more heavier soil, like a silt loam, where they were only able to move up 6 inches. So knowing where your nematodes are is really critical if you want to be successful in managing them. This is especially important if you realize that the current fumigants are much less volatile than methyl bromide used to be. This figure here shows you the vapor pressure of these different fumigants. It's very high for methyl bromide, which means that methyl bromide is very volatile. It basically, when you inject it in the soil, it will race through the soil. This is not the case for the fumigants that are used now. They're much less volatile. So especially to the deeper soil layers, they're gonna diffuse much less effectively than methyl bromide did. This is nothing new. Back here in the 1964, this is Lambright, who was a scientist working with Dow on Telone at the time, already was experimenting with these deep placement of fumigants, trying to get the fumigant deeper in the soil where a lot of the nematodes are hiding, and he found some improved efficacy with it. So right now in Florida, there's a lot of fumigation equipment optimization that's been done the past 10 years, let's say, by Joe Noling. And this is just a quick overview of all the types of equipment that are being used now to inject. And you can see some of these deep shank rigs, they can go 18, 20 inches deep below the hard pan, which is very common in our vegetable and strawberry fields, to inject the chemical where the nematodes are hiding. So fumigation is still really very common in Florida and is still the standard nematode management practice, especially for high value crops in our state. Obviously, there's problems with fumigants, otherwise I wouldn't be talking about it. They are expensive, they require special equipment, they're quite toxic, so you need licensed applicators, you need proper PPE, there's buffer zone requirements. This is a really big deal in California, but also in Florida, it's becoming more important. There's groundwater restrictions in certain places. They're certainly not a popular management tool. There's a lot of regulatory, societal pressure, to ban the use of fumigants. And they do cause collateral damage. You got to keep in mind fumigants are biocides, so they will impact plant parasitic nematodes, but they will also impact beneficial soil organisms, and as such may impact natural soil suppressiveness and long-term soil health. So I don't want to get into this too much because I know Christina already talked about it, but soil harbors an incredible diversity of organisms, that make up the soil food web. Christina mentioned some of these non-plant parasitic nematodes. So you have bacterial feeders, fungal feeders, predatory nematodes, but you also have a lot of these parasitic fungi, nematode trapping fungi, even predatory mites that will feed on nematodes. And I have some pictures here. You see, here's a nematode. This is a predatory nematode that actually grabs and swallows prey. This is another type of predatory nematode that pierces them and feeds like that. Here you see these spores. These are Pasteria, bacterial spores that parasitize nematodes and kill them. And then below here is 2 fungal antagonists of nematodes. So there's a lot going on in the soil. There's a lot of microbial antagonists of nematodes. There's a lot of different mechanisms also. This is a slide from a paper from Topalovic, a recent paper. I don't want to get in detail here. I just want to point out that all these antagonists, and there's different types of them, they can affect all stages of the nematodes, whether it's the vermiform stage, whether it's the egg, whether it's the adult stage. And some, may affect nematodes by trapping, by making these rings, these elaborate networks that trap nematodes. Other may be endoparasites. Some may go with parasitized eggs and females. You have bacteria. There's a lot of bacillus species that may produce toxins and may induce systemic resistance in plants. The same goes for a lot of the rhizobacteria. Some of those even produce volatile organic compounds that almost act like a fumigant. So there's a lot going on in the soil. For a nematology lab, looking at all that is just not possible. It's just way too much work. So what we do, and what Christina showed earlier, is we look at nematode feeding groups as indicators of soil health. So they're fairly easy to identify based on their mouth parts. A plant parasite, as I said, has a stylet, and based on the various mouth parts, you can fairly easily separate them into bacterial, fungal feeders, plant feeders, predators, and omnivores. So this is some data from a trial we did on strawberries, where we looked at KPAM, so it's a common fumigant in Florida. We looked at the effect of KPAM versus no KPAM on, in this case, fungal feeding nematodes. And we did this early in the season, mid-season, and late season. So early is about a month after fumigation, mid was about after 3 months, and late was about 5-6 months after fumigation. So you can clearly see an impact on these fungal feeding nematodes by the fumigant, especially early on. But even mid-season, there's still a highly significant effect. It starts to reduce a little bit when you go to the end of the season, but at least in this case, the effect, the reduction in fungal feeding nematodes was really throughout the season. This is another fumigant, C35. So this is a mixture of chloropicrin and talone, again on strawberry, and again early season, bottom, mid-season, late season. And it shows a similar thing. It seems that the fungal feeding nematodes are mostly affected by the fumigants. We do see some minor effects on bacteriivores, although their life cycle is really short, so they can bounce back more rapidly. And we also see an effect on omnivores here, but they're usually, their numbers are lower, so it's harder to see effects there. But clearly the fungal feeding nematodes are the ones that seem to be more sensitive to these fumigants. So, you know, you can call that collateral damage to beneficial nematodes of fumigants. But another thing that we often see following fumigation is that it increases the end of season plant parasitic nematodes while reducing the nematodes. What I mean by that is, if you look at the figure here on the left, this is the nematode damage, the root damage, with PIC 100, which is chloropicrin, PIC 60, which is chloropicrin plus talone, and you see a clear reduction in damage to tomato in this case versus the non-fumigated. But then when you look at the soil populations at the end of the season at the same time, you see that they're very high in the PIC 100. So while protecting the plant, it did not reduce the nematode populations. And then when you do a double crop, you know, a lot of growers, by saving money for plastic and drip tape, will double crop on their plastic. In this case, we double crop the cucumber. You can see that the root damage on that cucumber now becomes significantly higher than the control in this case. So what happens here, we believe, is that chloropicrin is a very good fungicide. So you know, it's good for reducing soil disease, but it will also negatively affect some of these natural antagonists of nematodes. And by doing that, reduce natural suppression of nematodes, which is why we see that rapid resurgence of plant parasitic nematodes after chloropicrin. So we did some survey work in Florida to try to figure out what is the natural nematode suppressiveness in Florida soils. This is published recently. This was done by my postdoc and myself. I'm not going to show you all the data. We just picked 4 sites here, 2 organic strawberry fields, 1 conventional, and 1 natural ecosystem. All very sandy soils, low in organic matter, except maybe the natural ecosystem was a little bit higher. And the microbial activity was measured, which correlates well with the organic matter, as you would expect. So as far as suppressiveness, and we do this by looking at differential gall development in steamed and untreated soil, you can see that we do get a good bit of natural suppressiveness in most soils, but clearly the conventional one, and this was a field that was fumigated for probably at least 30 years, the natural suppression is significantly less than in the organic or the natural ecosystem. We don't really see a difference between organics, agricultural soils, and natural ecosystems, which is interesting. We also did a high throughput genome sequencing. This is Sarah Strauss, who's now at UF, who used to be in California, I believe. And we do see a good correlation with fungal diversity, especially which again seems to be significantly reduced in the conventional fumigated soil. So, new nematicides. Just briefly, I want to give you a brief history of nematicides here. The fumigants were really the first nematicides that were out there, and they go back to the late 1800s. And as you can see, the fumigants that we use these days have been around for a long time, so there's nothing new about them. A second wave of nematicides was started in the 1960s and 70s. These were basically insecticides that turned out to have some nematicide activity, and these are your organophosphate and carbamates like Nemicure, Temic, Vidate, Mocap. Most of those are gone now. Some of those like Vidate are still being used. And then more recently, there's a new wave of nematicides. They're the fluorine-based nematicides, and they're all much less toxic and more selective. So that's a good thing. So this is just a table with some of the new products and compared with the old products and some of their properties. So Nimitz and Vellum are two new nematicides that are starting to get more commonly used in Florida now. Fluensulfone and fluoparum is the active ingredient. Another one here, Celebro from Corteva, should be registered later, probably in 2021. And then Nospirotetramat Movento is used in California. Mike McHenry did a lot of work with it in perennials, but we're not really using it in Florida as a nematicide, just as an insecticide. Anyway, I've color-coded them green just to emphasize their safety. Their toxicity category is a caution, just a caution label. So this is the first time we have nematicides that just have a caution label. Previously, they all had a danger label. They have new modes of action, which is good. So you can start rotating now nematicides if you like. One of the key differences is their solubility here. They're much less soluble than something like a Vidate, which is a good thing in the sense that you're less likely to get groundwater contamination. The negative of it is that it may be more difficult to get a uniform distribution in your soil when you apply it with water. They do have a little bit longer half-life, especially fluoparum, which is an extremely long half-life, so it's quite persistent. Fluoparum, as some of you may know, is also a fungicide, was actually first labeled as a fungicide called Luna. Later they figured out it's a nematicide, but so it's an SDHI. Yeah, so. We also do some work with biological nematicides. Some of the growers use them, often, in combination with conventional nematicides. Some of those are essential oils like thyme oils, like based on azadirachtin. Magistine here is toxins produced by Burkholderia, but it's so not a live organism. Melacon is a live organism. They're one of those nematode antagonistic fungi. And then there's some mixtures of mustard oil and capsaicin. So just some data on these fluorine nematicides that we worked with. This is one of the first trial. This is root damage on tomato. You see the untreated check here on the right. This is over time. And KPAM, right, new nematicides and combinations thereof looked actually pretty good. So they give a good reduction in root nematode damage. In some cases, just as good as the fumigant. This is another trial where we also had some of the biologicals in there. Here we looked at them with or without fumigants, sorry. So we had whole rows fumigated, and then we superimposed these products on top of that. The reason we do that is because, as I mentioned, these nematicides are much more selective. So they will not give you disease control. They will not give you weed control. So to compare them in a more fair manner, we decided to do it with and without fumigants. If you look at the gall, the root damage again, there's some variation. Nimitz seems to be the most consistent in reducing root damage. So it's the only one that really in the non-fumigated did something. Vidate looked good after the Pichlor 60, and then after the chloropicrin, all of the three new nematicides look pretty promising. So there's some variable control here depending on whether or not you fumigate, and that's something we're still working on and trying to understand better. If you want to rate them, as far as the fumigants, Telone is your most effective nematicides. Capam has a good broad spectrum, so it's a good nematicide, not quite as good as Telone, but it has a better spectrum than Telone, so it also has good disease and weed control. Chloropicrin will give you some nematode control, but as I said, you will often see rapid resurgence of nematodes, but it is a very good soil disease product. As far as the non-fumigant nematicides, all of the chemicals we tested look pretty good on root knot, unless the nematode pressure gets very high, and then you may have to add a fumigant to knock them back. As I said, no disease and weed control. So, you'll have to, if you don't want to fumigate, you'll have to consider that and look for appropriate fungicides and herbicides if possible. Right now, we're looking at mixtures, trying to mix some of these products and see if multiple applications can give us some benefit. As far as the biologicals, you can get anything from poor to good nematode control, but typically you need multiple applications for the biologicals. And mixtures with chemical nematicides seem to really improve efficacy in most cases. One major benefit that we saw from these new nematicides is that they do not seem to have much of a negative impact on the beneficial nematodes. This shows you the different feeding groups. Capam here on top, as I said, really knocks back the fungal feeding nematodes as well as the omnivores here. Not so much the bacteriivores, but if you look at the other non-fumigant nematicides, they really don't look too much different from the untreated control. So that certainly is a benefit over fumigant nematicides. Biological management. This slide just showed you, a whole range of different options you have if you want to biologically manage nematodes. I'm not going to get into all of this. This would take too much time, but I'm going to touch on some of it. The first one is really, it's a no-brainer, but it's often forgotten. It's good agronomy or good crop husbandry. Plants are less likely to respond to nematode management if other conditions are limited. And this is really based on Liebig's law of the minimum, which was actually Carl Sprengel that came up with it. But if you look at this barrel, it's often shown by this barrel, the amount of water this barrel can hold is really limited by the shortest stave, right? So, and it's the same for nematode management. It doesn't do you much good to optimize your nematode management if you don't take care of other adverse growth conditions like drought stress, nutrient deficiency, certainly other pests and diseases and such. Sanitation, a major key issue, obviously, you always should make sure you have nematode-free transplants or rootstock, especially when you move into new fields, virgin territory. It's critical that you try to avoid introducing nematodes. There's a great case study for the citrus industry in Florida. This was Larry Duncan told me this. He's our citrus nematologist. In the 60s, 70s, you had, these two diseases were the main issues in citrus: slow decline caused by citrus nematode, spreading decline caused by burrowing nematode. So in response to that, Florida implemented a rootstock certification program in the 70s. Once that was implemented, this really drastically reduced the spread of spreading or slow decline of Tylenculus. And the impact of that became clear in the mid-80s and early 90s. The citrus industry in Florida suffered some severe frost damage, and all of the groves shifted more south. So they basically moved more south. Because of this certification program, we hardly see any citrus nematodes in the south. So we were able to prevent the introduction there by having this certification program. Host resistance is probably the most effective economical method for managing nematodes, both in high and low value cropping systems. Soybean cyst nematode in the Midwest is almost entirely managed by resistant cultivars varieties. Tomatoes, processing tomatoes in California are to a large extent managed with resistant varieties. And as far as perennial crops, in citrus, there is some good rootstocks that are resistant to citrus nematodes. They've been around for more than 25 years. But we do see some virulent patatypes showing up recently. And that's, of course, one of the drawbacks of resistance if that's the only thing you rely on. There's some root knot resistant tolerant grapevine rootstocks available. And I refer to this fact sheet here again from University of California, where you have a nice table listing all the different rootstocks available. Probably the best example of resistant rootstocks is for prunus crops, and the Nemagard rootstock has been used for more than 30 years, very successful, no resistance breaking issues, and it's used in most of the almond, nectarine, peach, and plums in California. There's also some resistant, root knot resistant walnut rootstocks available. So you notice that most of the resistance is going to be for nematodes like root knot endoparasites. It's much more difficult to find resistance against ectoparasites. I believe there's some resistance for dagger nematodes on some grapes, but that's one of the few exceptions. One note here also is that when you introduce resistant root knot rootstocks in your grove or vineyard or orchard, it's best to do it when you replant the whole grove. If you replace just some resistant rootstock next to infested susceptible trees, you really have a much higher chance of developing resistant breaking biotypes. Cover crops for nematode management. I'm not going to talk too much about cover crops because I think our next speaker is going to focus on that. But we're doing quite a bit of work looking at that. This is just the list of the crops that we've looked at. Some of the more common cover crops in Florida are sun hemp, southern peas, and different sorghum Sudan grass varieties. So this is some data from a greenhouse trial that we did where we looked at all these cover crops and we looked at them for 3 different nematodes: northern root knot, northern lesion, and sting nematode. These are the 3 nematodes that are important in strawberries, and they can all cause damage. And you see, I kind of color coded it so it's easier to read. Strawberry is our crop, so this is a good host for all these nematodes. But you see, it's very hard to find crops that are green throughout. And the ones that are here, sugar beet, really didn't grow well. The same with goat's rue. So on the crops that really are important for Florida, like sun hemp looks pretty good. It's a poor host to root knot, and it is for most root knot species. Also for sting, but it is a pretty good host for lesion. So that could be an issue if you have lesion. Sorghum Sudan is common in Florida, usually a poor host in most root knot species, but it is a very good host for sting. So for strawberry fields, it may not be such a good cover crop. But basically, I think what this table shows that it's, if you have mixed nematode populations in your field, it is very complicated to find a good cover crop. So how do you manage mixed nematode populations? Well, one way to do it is by mixing your cover crops as well. This is some data from my earlier PhD work when I was back in Kenya, working with cover crops. So the cover crops we worked with there were, these are all legumes, Tephrosia, Sesbania, and Crotalaria. And Tephrosia and Sesbania were good hosts for root knot. Crotalaria, as I just showed, is a poor host to root knot. So when we combined the cover crops as far as mixtures, you see when you combine 2 good hosts, it really doesn't make a difference. The blue bar here is the nematode population, the root knot nematode population. It's still about the same. Crotalaria is a poor host, so you get really low populations. When you mix a poor and a good host here, Tephrosia and Crotalaria, you do take down your absolute root knot populations, but you still get fairly high populations. That's why I put the orange bars there. The orange bars are your relative populations. So this is a root knot population relative to your total plant parasitic population. We had a lot of lesion, a lot of spiral nematodes in these soils, which are not as damaging to the crop that we were focused on, which was bean. The picture on the right shows you the typical intercrop there, which is maize and bean is especially susceptible to root knot there. So when you look at this relative population, you see that this is much lower now in this poor and non-host mixture. And that really seems to be what more determines the yield, which is in these gray bars here. So you get much better bean yield with the poor host and the poor and good host combination, because probably the relative population, which is more indicative of the actual damage that root knot can do on the total bean crop. Another tool you can use as far as cover crops is using certain cover crops for biofumigation. Some work we did when I was working in Tifton, University of Georgia. And I just want to show you the process, the idea behind it. So brassica cover crops like mustards, like radishes, they all contain these glucosinolates, which are basically sugars with a sulfur on it. When you break those down, when you chop those up and you add water, they turn into glucose and a whole number of toxic compounds, of which isothiocyanates are one of the most common. And this is the same as the active ingredient in products like metam, capam, and basamid and such. So the way we did the work there, we grew these cover crops, these brassicas over winter. They do quite well over winter. We mow them early spring. You get really good biomass. Rototill them in. We use this flail mower, which really chops them up nicely. Till them in, add some fertilizer, irrigate them. We didn't have drip irrigation there, so we used overhead because you really need a good amount of water to hydrolyze these materials. Made plastic beds on top, and then planted our vegetable crop. Results were kind of mixed. Radish, I believe, was the best looking. One of the problems with some of the brassica crops, if you grow them for nematode management, is that some of them are actually good hosts for certain nematodes. So while you grow them, you may actually be increasing your population. I know there's a lot of these brassica varieties out there that have really been bred for high glucosinolate content, and so hopefully in the future there'll be some varieties that may work in different regions. This is some work that Tristan Watson, who was my former postdoc, he's now a nematologist in Louisiana at LSU, did for his PhD, where he was looking at compost and mulch for nematode management. So this was in Canada, in Summerland, where he was working on sweet cherries, and the nematode they were dealing with there was Pratelinchus, lesion nematode. So the treatments he used were fumigation, basamid, which is like a metam ITC generator, compost, bark chip mulch, and the combination of both. And this is the pictures kind of show you how they did it. This shows you the effect that these treatments had on trunk diameter of cherry trees. They measured over 4 years. So the first year you see the fumigant in orange stands out. So you get this short, quick burst of growth after the fumigant, which you don't see much with the other treatments. But even the second year, you can already see that some of the, especially here, the one in blue, the compost and mulch start catching up. And over time, you see gradually the compost treatments start actually looking slightly better than the fumigant treatment. The fumigant is still looking better than the control, but you know, certainly doesn't look better than the compost treatments anymore. So Tristan also looked at the lesion nematode root infestation. This is nematodes per gram root. You know, there's a lot going on here. There's a lot of seasonal fluctuations here, obviously. But I just want to point out here the dark blue, which is the compost plus bark mulch. And it's the curve that's really always kind of at the lowest bottom. So you seem to maintain fairly low populations with the mulch and compost. The fumigated soil here in orange, what it really shows is that you get a rapid reinfestation of Pratelinchus, which is something that we often see in Florida also after fumigants. They also measured soil suppressiveness, like I mentioned earlier, similar assay, but here they looked at differential recovery of juveniles of root knot from steamed and unsteamed soil. And again, you know, just point out, this is suppressiveness percentage. All the compost or bark chip mulch really have clearly higher suppressiveness than the control and the fumigated ones. Some other soil amendments that we've done some work with. This is a biodiesel waste product called Verdesoil. It's basically glycerin. It's from a company, a big biodiesel company out of Iowa that did some work with it in Florida a few years ago. I'm not sure they're still working with that product, but This is some work we did where we compared it with a fumigant PIC 60. We applied the glycerin material as a drip and through the sprinklers. And the data you see is the strawberry yield in blue and the sting nematodes in orange. So you can tell the yield, if you look at the orange bar, the yield in blue, I'm sorry, is increased over the untreated with the Verde soil, and especially the sprinkler application looks almost as good as the fumigant. As far as sting nematode count, we don't see a similar reduction than PIC 60. We do see some reduction, but clearly the material can improve strawberry yield, there's no doubt. Some other uses of organic waste is for anaerobic soil disinfestation. I personally haven't done much work with it. I have a colleague in Florida, Edwin Ruskopf with USDA, who's done a lot of work on it. People in California are working on it as well. So what's ASD, or anaerobic soil disinfestation? It's a pre-plant, non-fumigant soil disinfestation practice. First started using this in Japan and the Netherlands early 2000. And it basically uses carbon sources. You can use dry, liquid, that provide a substrate for proliferation of anaerobic bacteria under saturated soil conditions. So you need to add a lot of water to it to stimulate this process. What happens is when you do this, is you change the soil microbial community composition. There's production of volatile organic compounds that may be nematocidal and fungicidal. And you basically generate lethal anaerobic conditions. This is some pictures of how they've been doing it. They make these false beds here for tomatoes. Then they add some chicken litter, and in this case, molasses. We have a lot of sugarcane in South Florida. So molasses or a waste product from the sugarcane production. You put it on these false beds, and then you add water, and you basically wait 2 or 3 weeks like you do with a fumigant before you plant. Solarization is not an essential component of ASD, but it's been shown that heating the soil when you do this can actually improve efficacy. Talking about soil heating, this is some work we've started recently looking at solarization. This is a rig that we inherited. You see we lay this plastic about 13 feet. It's clear plastic, so you basically heat the soil. This equipment has some drip tapes underneath, so we can keep the soil moist, create more of a steaming situation, and we can even inject other products underneath it to make it more effective. Another way of heating the soil is steaming. On the left, some work that was done in Florida where you actually put the steam directly into batch using this boiler here. The one on the right, there's a lot of work being done in California by Steve Fenimore, University of California wheat scientist. This is, I think, one of his latest rigs that uses spikes, 10-inch spikes that inject the steam into the ground. Of course, one of the drawbacks of steaming, it is expensive. You use a lot of fuel. I think the equipment has been getting better and better and more efficient. So at some point, we may come to a point that it's actually becoming economically feasible. So finally, and this is my last slide, just some key points I want to make before I take it to questions. But it's really important, and I think that's something that's been lacking, to develop regionally relevant data to control nematodes. You know, even here in Florida, we see what works in one region may not work in another. So you can't really generalize when you talk about nematode management. Better knowledge of nematode biology will improve management, and you always deal with the diversity of nematodes to manage, which definitely complicates things. Long-term field trials are needed to evaluate especially some of the more sustainable nematode management practices, to figure out what's the short-term versus the long-term benefit, which is definitely critical when you're dealing with perennial crops like grapes. And nematode resistance really should be a priority for plant breeders. Unfortunately, nematodes are usually not on top of the list of plant breeders, but hopefully that will change in the future. And with this, I'll take it to questions if there is some time. Thank you.
We've got the mic, Dr. Dessiger, so I'll ask you a couple questions right off the bat, if you don't mind. Thank you very much for the presentation. That was fantastic. A lot of new information, for us in the wine grape industry. In terms of the efficacy trials for the different nematicides that you showed us, the depth that those samples were taken at, is that all like top 12 inches? Based
Yes,
on
pretty
crop?
much. Yeah, those are all pretty much 10, 12 inches. Yeah.
So I've been interested in KPAM for a long time, and you know, I think there are some complications or challenges to applying it in vineyards, but it's doable, and people have done it. One of the benefits, it seems to me, be the you know, you're putting out a fairly significant amount of potassium based on the application rates required for success.
Right.
And so it looks to me like, you know, if you ran it pre-plant, that would essentially, you know, you could get some dual benefit. Like you're putting out, you know, grapes require a fairly significant amount of potassium in terms of the, you know, the plant need. And so you're achieving two things. You're cleansing the soil, you know, within whatever range you can get the material down to, but you're also picking up a pretty good shot of potassium, right?
Right.
Cool.
Yeah, no, so KPAM, I know, and you may want to check with Inga Zasada at USDA. I know she has done some Metam KPAM injections in vineyards up in Oregon. And she told me she's really seen good control of dagger nematode, especially. And, you know, over several years, not just the first couple of years, but over time, it seems to do a really good job on dagger nematode. Root knot may be a little different, but I think KPAM's pretty easy to apply. You can apply it through a drip. It's a lot easier to apply than things like tallow and chloropicrin, for sure.
Thank you very much.
Let me see if there's some questions here. So there's a question, what has your experience been with Diterra? I unfortunately have no experience with Diterra. I've known other people that worked with it, but I think it's been pretty much like the other ones. There's another question on Diterra, Nemaque. I haven't worked with Nemaque also. I know it's been used in California. We don't really use it in Florida. Done some work with Melocon in the field. It's been mostly disappointing. Sometimes we do see some better effect in when we do it in a greenhouse in a pasteurized soil. Melocon, you know, is a live organism, so it's a live fungus. So when you put it in a live soil, it has to compete with all these other organisms, and that may obviously impact its, you know, its capacity to colonize. Right now, we're looking at seeing if we can combine different biologicals and make them work better as such, and that seems to have some promise. But really, when you use them as individual products, they've been, at least in Florida, fairly disappointing. Then there's another question, do the biofumigants affect non-plant parasitic nematodes and other parts of the soil microbiome? That's a good question. And some of the biologicals, they do. Certainly the biofumigants, I think they can, probably because the concentration is much less than a chemical. It's probably not going to be to the same extent. But we have seen some of these biological nematicides that are based on essential oils may be able to knock back non-plant parasitic nematodes just as well. So yeah, that's a good question and something I think we need to do more work on. I see another question on biofumigation. How essential is the irrigation aspect of biofumigation? If you can irrigate, does that increase the effectiveness? Same question about plastic beds. Yeah, I think for biofumigation, I really think that irrigation is a key aspect of it, because the whole breakdown process, the degradation from glucosinolate to isothiocyanates is a hydrolysis, so it needs water. So unless you have already good moisture in your soil, if you can't irrigate, you may want to wait till you get a natural rain event before you do that. And then there's another question on Diterra and Nemaque. as I said earlier, we haven't seen much results with it here in Florida.
All right, just to stay on track, if you don't mind, Dr. Segura, if you would stay online and catch any questions that pop up and answer them via the chat function.
I'll be around for a while, so I'll keep an eye on it.
Awesome. Well, thank you so much again for the presentation. That was fantastic, and we all learned a lot.
Thank you.
It was wonderful to have you join us.
Appreciate it.
Thank you. And then, so moving forward again, keeping with the theme. Actually, I want to point out one thing before we move on to the next speaker. Every year we open this series of trainings up free to students, and I want to say hi to all the students that have joined us today. We've got it looks like 10 plus different folks joining us on that level, from institutions like Napa Valley College, University of California, Berkeley, UC Davis, a number of people from Berkeley. Additionally, we have someone joining us from Washington State, so yeah, a pretty good reach. And as attendees, please spread the word about these trainings between now and the last one to anyone who is a student and who might benefit, because they will have free access. Okay, moving forward, our next speaker, who has joined us before, Dr. Anil Shrestha. He's a wonderful guy, and very knowledgeable in the area of weed science. He's the chair of viticulture and enology at Fresno State, and he holds his PhD and master's degree from Michigan State in crop ecology. I'm sorry. His PhD is from Michigan State. His master's degree is from Cornell, both focused on crop ecology. The presentation that he offers us today is going to be called Cover Crop Crimping, Practical Application in Vineyards. This is something that I truly believe will be a huge part of the future of weed management in vineyard systems, and it's something that I personally have had great success with in Monterey County, and it's not cutting edge stuff in other crops, but it is in vineyards at this point, and really looking forward to this presentation. Without any more comments from me, Dr. Shrestha.
Well, good morning. I hope you can see my screen. Is that okay, the screen?
Yes, we can see it.
Okay, thank you. First of all, I'd like to thank Eric and the Napa Farm Bureau for this invitation. And as Eric said, I'm based at Fresno State, and I'm a weed ecology person, and I've been working on vineyards for quite a while now. What I'm going to do is share some experience with cover crop crimping, and this is mostly from an agronomic standpoint. In an agronomic crop, corn was the crop that I was working with, and I have data for that to share. But I think it has quite a fair bit of potential for vineyard systems. And as Eric was saying, he's already doing some of this work in a vineyard setting. And I'll try to present as much data as possible from my own experience, so you can see the connection between a corn system and a vineyard system. So I'll try to make it as relevant as possible. Having said that, I'm going to begin with some very basic information on cover crops, because I just heard that we have students in this panel, and I'm glad that the previous speaker covered a lot of information on cover crops as far as nematodes go. So when we talk about a cover crop, we are talking about a crop that is primarily used to cover the soil for protection from soil erosion and nutrient losses. And it's usually between 2 crop growing seasons in agronomic production systems. But in trees and vine systems, we're talking about putting cover crops in between the tree rows or the vine rows. And primarily it's a non-economic crop grown in the vineyard middles. And occasionally in my own PhD work, I was looking at whether we could harvest a cover crop and get benefit as an animal feed and still get the other benefits as cover crops. And I did some work on that before. But here I'm going to talk about non-harvested cover crop and how we managed it. And in some instances, it's also used interchangeably with the word green manures. So if you look at the history of cover crops, it's not new at all because this started way back in 5th century in China, and then Rome, farmers were also using cover crops. Then green manuring was used in England in the 18th century, which was basically cover crops. And then early on in the 20th century, cover crops became more common in North America. But once the synthetic fertilizers started becoming available after the Second World War, the use of cover crops kind of declined. But in the last few years, or I should say decades, from the 90s or late 80s, 90s, there has been a lot of interest in cover crops, and most of it has to do with the ecological benefits that cover crops bring with them. So when we talk about cover crops, there are several benefits, and we already heard about suppression of nematodes from the previous speaker, but we are looking at adding organic matter and in the process sequestering carbon with cover crops, reducing erosion, increasing water infiltration, decreasing nutrient loss. And if you have a nitrogen as a cover crop, as a legume, as a cover crop, you can fix nitrogen. And then weed suppression is a big part of it as well. And I have a personal interest being in weed science, looking at the weed suppressive abilities of cover crops. It increases mycorrhizae and also attracts beneficials. So if you look at the summary, it's like improving the soil health, and that's why there's so much interest in cover crops. I've been working with a group of farmers in the Central Valley, plus actually it's all over the state. We have about 6 organic growers on a grant supported by NRCS, and we're looking at the kind of information I'm going to present today on the roller crimper part of cover crops, but they're primarily working on vegetables, and they are all organic farmers. But in my case, I was working with corn. So cover crops add organic matter. So that's one of the benefit that we have from cover crops. And we've also heard of carbon dioxide increasing in the atmosphere, and it's like around 420, 30 at that level now, I think. That's the data that we get, and it's been a rising trend over the years. And carbon dioxide is one of the most commonly produced greenhouse gases, and agriculture is also considered to be responsible for some of the production of carbon dioxide. So we look at methods of sequestering carbon and storing it in the soil, and that process is called carbon sequestration. So it's one of the methods of reducing the amount of carbon dioxide in the air with the goal of reducing greenhouse gases. So when you plant a cover crop, it's fixing carbon dioxide by photosynthesis, it's storing it in the biomass, and then the roots are also the root exudates also contribute to the organic carbon, and it goes to the soil. And when this cover crop residue decomposes, and both the above and the below ground biomass decomposes, it stores carbon in the soil, and that prevents it from going back to the atmosphere as CO2. So cover crops have quite a big role in sequestering carbon in the soil. So we have another study with some of my UC colleagues at the UC station at Five Points, California. We've been looking at cover crops and no-till systems for almost 20 years now. And the experiment was set up with 2 factors. We were comparing standard tillage and conservation tillage. and with and without cover crops. And we were rotating cotton and tomato in these plots for 20 years. Now we've switched to sorghum and garbanzo bean in these same plots. And there's some information that we have generated from that study there that kind of adds to or supports the benefits that I was just talking about earlier. We have found that over the 20-year period, this conservation tillage with cover crop, it added almost like 15 or more than 16 tons of carbon in the top 6 inches of the soil. And the conservation tillage with no cover crop came in second, whereas the standard tillage systems were less in terms of soil carbon than the top 6 inches of the soil. So that's a study that kind of supports what I was displaying earlier on the benefits of carbon of crops. Then there are studies showing erosion, reducing erosion, the benefits of reducing erosion from cover crops. And we also found from our own study at 5 points that with conservation tillage and cover crop, the wind erosion was almost zero compared to the standard tillage with no cover crop. The wind erosion estimate was about 2.1 megatons per hectare. So cover crops definitely have a role in reducing soil erosion. And the other benefit is increased water infiltration. So this is a slide from Salinas area, I guess, Monterey County, somewhere there. So when you have cover crops, the water infiltration is pretty high and you don't really see runoff after a big storm event as compared to a plot with no cover crop. There's water logging, and then there is quite a fair bit of runoff from these vineyards. And our study kind of verifies that. What's happening is, if you look at this data here, the standard tillage with no cover crops or cover crops, the time it takes for the water to infiltrate is very high. These are in seconds. Compared to the no-till with cover crops, you can see that the water infiltration is very quick into the soil. So that kind of supports the views that you see here, that the water infiltration is pretty high in no-till with cover crops compared to standard tillage with or without cover crop. And I'll get back to this again when I show you the soil structure of soil with cover crops and no cover crops. Then there are studies that show that cover crops reduce the loss of nitrogen and phosphorus to waterways. And we did not document, or we did not study these things in our study, but there are evidences showing that. And of course, soil nitrogen fixation from legumes is well documented, and there are a lot of studies showing the amount of nitrogen that is fixed by these legumes that you use for cover crops. Weed suppression is a big one, and this was a plot in Hollister, California, where an organic grower uses cover crops a lot to suppress weeds. And when you have that dense of a canopy of cover crops, it's very rare to find weeds in there. And this is from my own plot here with cover crops. And when we terminate the cover crops, this is what we see. And there are almost no weeds. So there's that benefit of saving on weed control or herbicide sprays, depending on what type of method you want to use for cover crops. For weed control, you get a lot of benefit from cover crops in terms of weed control. And the other data that I found was that cover crops can boost soil mycorrhizae, and which can lead to reduced inputs. This is from South Dakota State, so they have documented that. And nematode management, I don't have to talk about that. The previous speaker completely covered a lot of that information, so I'm not going to go there. The other benefits are attracting beneficials, providing shelter and nectaries to these different butterflies and bees, and this can also assist in pest control of other pests as well. So this is from Glenn McCarty. His data have shown some of these information, and this is again from him as well. So there are dedicated insectary rows that has been used in vineyards for um, benefits from cover crops. And then the big part of all this is enhancement of soil quality and soil health. So you're adding a lot of organic matter, improving soil structure, and increasing the porosity of the soil, and also lowering the bulk density with more carbon. And you're also providing food source of, in the terms of carbon for soil microbes, and this improves ecological cycling. And this is not going to happen overnight. It takes a while for things to happen. But in our study, we've seen that, you know, when you have cover crops in the system, and the ups and downs you see is primarily because of the rainfall pattern. 2007 was very dry, so we ended up with almost no cover crop biomass, and we tried to minimize our irrigation as much as possible. The only irrigation we put on, depending on the need, was during establishment. The rest of the season, the winter season, we tried to not add any irrigation at all because it adds to the cost. So over the years, we've added almost 32 tons of organic matter in the systems that that I showed you in the 5-point study, and that equals to about 15 tons of carbon over the 15-20 years. So that's a lot of carbon that we've added in the soil. And you can see the quality of the soil with the cover crops and without cover crops. So there's quite a fair bit of difference, visual difference as well. But this was the most interesting thing that Dr. Jeff Mitchell, he documented. On one side of the screen, you will see soils that have been under conventional tillage without cover crops. And on one side, you'll see the soil that has been with cover crops for a long time. And notice like what happens when you put the soil clods into the beakers or jars of water, and how they break down over the time. So I don't think I need to put the sound on for this because it's more evident. So this is what happens when you have a soil that has no cover crop in it. That's a standard tillage. Look at how fast it's breaking down, and compared to the ones that have the cover crops. So this is the one with the cover crop, and it doesn't flocculate. And this is why we see, you know, a lot of water holding capacity build up and no runoff, and the soil structure kind of improves over time. So this I found was a very good demonstration from Dr. Jeff Mitchell, showing the importance of the cover crop in improving the soil quality, soil health, and also the soil structural ability. So this one's completely flocculated, and you can imagine the amount of erosion and runoff you're going to have from that type of soil compared to the one with cover crop. So that's a nice demonstration that Dr. Mitchell has from our 20-year study. And the other thing that we have been looking at, and I don't have data to present here, is we are also looking at the diversity of various critters in the soil, earthworms and insects. And we have evidence to show that the plots with cover crops have more soil insect diversity, and also earthworm population is higher in the soils with the cover crops. And I wish I had the data prepared for that, but we don't have it, but we have evidence of that, and we are in the process of analyzing that data. The other thing is with cover crops, some people say that cover crops kind of spoil the aesthetics. I think it improves the aesthetics, in my opinion. And there are drawbacks with cover crops. The big one would be increased water use in a arid situation like ours, where we don't get a lot of rainfall, competition with crop nutrients, frost hazards. In some cases, we've seen increased diseases and pests. And also there's that economic part of increased costs for management and additional equipment that may be required to manage cover crops. And for some people used to seeing clean fields, it may be an issue of aesthetics. So we looked at, this is again from the 5-point study, when we had cover crops in the system, we had a negative water balance. So if you're in an annual crop system or even in a perennial crop system, this is fallow. So in fallow, we were storing quite a fair bit of moisture in the soil, but in the ones that had the cover crops, we had a decrease in the soil moisture level in the soil profile. So that's a point that you may have to think of. And the other thing, if soil temperature is of concern, the cover crop kind of warms up the soil a little later than the bare soil. So the cover crop tends to keep the soil cooler for a longer time, and it can reduce soil temperature by 5 to 8 degrees Celsius here in the Central Valley. And that could have implications for agronomic crops that are planted in early spring or even in a vineyard. If soil temperature has an effect, it could affect the vine growth. We don't really know about what the effects are, but we are just assuming that some of these parameters may affect the crop growth. Now, when we talk about cover crops, we're talking about winter annuals, summer annuals, and also perennials. And mostly we use legumes, grasses, mustards, and sometimes people just use weeds as natural cover crops in some of these vineyard systems. So these are some of the common cover crop species: winter pea, berseem clover, bell beans, and vetch. Some of these are legumes, some are not. Mustards, so that's another common one. Phacelia. Personally, I like Phacelia in the system that I'm working with in terms of roller crimping, and I'll show you some data on that. Rye is very popular if you want great biomass. That's the popular one in the cover crop systems here. Oats is another one. Then triticale. And it also depends on how much tall height you want in these cover crops. Rye seems to grow the tallest. Then fescue grass has been used in vineyards as well. And mixtures, there are several mixtures that you can buy. And they're like, this is the one that I have been using, one called Multiplex. And I'll show you the content in that. And also, this is from Glenn, again, data showing mixtures of peas, veg, bell beans, and oats as cover crops in vineyards. Tillage radish is another one that's used mostly in the agronomic systems. They seem to be using tillage radish as well, or just weeds. And this is from our campus at Fresno State. We just use resident vegetation as a cover crop system, but we also are trying to explore cover crops in our vineyards. So when you think about cover crops, the important factors to consider, number one is seed cost. Some of these cover crops are pretty pricey, especially the mixtures can get to quite a fair bit of, add to a fair bit of cost. Then drought tolerance is the big one. You want to have a cover crop that does not require a lot of water. Cold tolerance, how much nitrogen do you want the cover crop to fix, or how much biomass, how much residue you want, how much soil cover you want. And then also the big one is easing termination. Can we terminate the cover crop easily or not? And the point that I make over there is many of these cover crops are terminated by the use of glyphosate or paraquat, so you may want to avoid that. Then when it comes to cover cropping, obviously prepare the soil and seed, and then if you have a legume, you need a rhizobium culture with it. And irrigation, it depends on rainfall. We have been irrigating only during the establishment phase, so we add about 1/2 inch, 1 inch of irrigation at establishment if there is no rainfall. And in fact, I just planted on Tuesday, that planting probably will require some rain. I mean some rain and irrigation as well. The forecast was for rain, but we didn't get much. Then the other question that comes is, can I have a self-seeding cover crop, or do I have to reseed every year? And I'll show you some data on that as well. So there are all kinds of implements for cover crop seeding depending on your situation. And if you're in a vineyard, there are special equipment for cover crop seeding in vineyards. And termination is the big one. So after you've grown a cover crop, what do you do to get rid of the cover crop? So one method is to apply an herbicide. This is from Firebaugh, where the grower had triticale as a cover crop, and the next crop is going to be tomatoes. So the grower applied glyphosate to terminate the cover crop. So that's a field showing application of after application of glyphosate. So that's one way. The other way is to disc and incorporate. And you can see that this one would require less soil disturbance. This one would require more soil disturbance because you're discing, incorporating the cover crop residue. And here's another picture of mowing and disking to incorporate the cover crops. And some of the, I found this from a website picture of terminating of cover crops. It's pretty neat, so I just wanted to share it, just not from my experiment, but just wanted to share it. And you know, when you till the soil, there's negative effects of tillage. When you till the cover crops in, you're losing organic matter, and you're also decreasing infiltration of air and water, and then the soil structure is being disturbed. And when you have any sort of disturbance, biological activity and diversity is also disturbed. So the other method is mowing. So one method is herbicide, the other one is disking, and the third method we have for terminating of cover crops is mowing. So these are like the three common methods of terminating cover crops. Here's a vineyard in Madeira where we were mowing the cover crops, and then we came back again and disked the plots. So just like the negative effects of tillage, there's also negative effects of mowing. You tend to have regrowth of these cover crops, especially if they're grasses. And if you don't want them to be regrowing, that could be a problem. And many times you select for weed species that are tolerant to mowing. Some of these low-growing species like knotweed and you know, puncture vine and those kinds of things, you select for those species if you have been mowing the cover crops every year. So here comes the roller crimper study, the research that I'm going to talk about now. And this is being used fairly widely now, especially in the organic systems. And I'm glad to hear that the Monterey Vineyard is also using this technology. So what it is water-filled drum with these chevron pattern blades that's attached to the front of the tractor usually. And we used it at the back of the tractor. So you can have a front mounted or a rear mounted roller. It mows the plants down and then it cuts the stems every 17 inches. So that's how it is designed. Of course, there are various models of these roller crimpers. And here's a video to show how this works. So instead of using herbicides or disking, plowing, we were just rolling the cover crops. The technology is called roller crimper technology. So this is from my plot at Fresno State that I'm now going to talk about. So this actually, we started with this technology way back in 2005 in 5 points where we were planting cotton and rolling the cover crop on the same day and the same pass. So you can see the front mounted roller in this instance. This was a triticale cover crop, and the equipment also was attached with a cotton planter. So we were no-till seeding cotton at the same time. This technology did not take off because to have the cotton emerge, we had to irrigate right away. And that was bad because once we irrigated after the rolling was done, the cover crop came back and it was a nightmare. We could barely find any cotton in that plot, and we gave up thinking, this technology is not going to work for us. That was way back in 2005, but now in recent years this technology has come back, and this is the same plot that I showed you being rolled in the not the previous one but the one before that, where we had used a big roller. And this is during corn planting, and we could easily strip till plant in that cover crop residue with a strip tiller. And we didn't have to use any herbicides, or we didn't do any disking or anything like that. We just went straight strip till with this machine and got our plantings done. So this is at Fresno State. This is under a centrifugal irrigation system that I am going to share now. The treatments I had, this was a replicated study. I've been doing this for the past 3 years. I just planted a few days ago, just 2 days ago actually. High diversity mix, Merced rye, purple vetch, other high diversity mix, Phacelia, rape, bill beans, Magnus, P, multiplex. So these are some of my treatments. And this one, high diversity mix, was pretty diverse. You can see there's almost like 20 different types of seeds in there. And this was the highest diversity mix that I had. So that's another shot showing you all the seeds in there. And this is one of the plots with the diversity mix. This one had bell beans, peas, and barley vetch. Rye was another treatment that I used. Phacelia, mustards, bell beans, and multiplex cover crop mix. This one had 3 different species, or 4 different species, I think. I have to look at the label. So for planting, we planted in December 18 and November 2019. And as you can see, just 2 days ago, 2020, that we planted. So again, we just planted with our planter, and we adjusted our seeding rates based on the biggest seed size we had in the mixture. So the seeding rates vary quite a fair bit. And then we had to irrigate with a ½ inch of water in 2019. 2018, we didn't have to irrigate at all. This year, I think we will have to irrigate. And over winter, you can see that these cover crops took off fairly well. Nice cover. This is Phacelia. Phacelia is one of my favorites, and I'll show you why in a moment. Peas, and the diversity that we got in the middle of Fresno in December is amazing. And you can see the beautiful Phacelia in the foreground, mustards, and this is another shot. In the middle of the city, it's pretty good to have these kinds of things. And there were visitors always looking at what was going on, all kinds of bird diversity. But the question is, how do we document these things, these insects and these, how do we keep a record of everything and present it as a benefit of cover crops is a question. So here's a aerial view of how my cover crop plots look like. And then this is again showing you the cover crop rolling crimping process. And again, we didn't have to use any herbicides at all. We just went one pass this way, and we decided to come back with a second pass on the same plot so that we could get a good kill. And I must say that the later you go with the rolling crimping process, the more success you may have. That's what my experience showed. If you go too early, what happens is you get regrowth, and then you have to deal with the second rolling, or even third or fourth rolling of the cover crops. And some of them don't die at all. And I'll show you some plots that had that problem. Most of the plots, they rolled very flat, and they kind of didn't regrow. This was the one that we had the most trouble with, the mustard, the rapeseed mustard. It wouldn't roll at all. It wouldn't, go down, and then kept coming back, and it grew throughout the season, interfering with the corn. So this is some of the things that you have to think of if you're in an agronomic system. If you're in a vineyard, probably this may not matter because it's not competing with the crop directly, although it may have some indirect effects. Even after the termination of cover crop, you can see the amount of visitors to the cover crop plots compared to the no-till plots. So there's the diversity factor. Here's the no cover crop plots, and here's the cover crop plots. The amount of soil cover we had over the winter, and the biomass. We got the highest biomass with the high diversity mix. And in 1 year, last year I have a plot just with rye to see what kind of biomass I get. And I got excellent biomass with rye alone as well. That is not in the slide, but just to give you an example of here, having almost like 16 tons of tons per acre of cover crop biomass with these systems. And you can see the amount of soil it covers. And the other thing that we have to think of in these systems is you want the cover crops to die, and also the residue to be disintegrating or decomposing. So the bell bean was the one that decomposed the quickest, and the rape, that didn't decompose at all because it kept growing. The others kept growing over not growing, but the residue was there on the surface. So the bell bean disintegrated very fast, and the phacelia was the second that disintegrated very fast. So it depends whether you want to have the residue staying for a longer time or you want it to be decomposing quickly. That's another thing that you have to think of. Now, as far as the killing with the roller went, all of the plots had good control with the roller, except for the rape, which kept growing, and we had quite a nightmare of planting corn and growing corn in this plot. Other than that, we had to use no herbicides at all in these plots, and you can see the amount of residue and the soil cover, and there are practically no weeds at all. This is phacelia, and the reason I like phacelia is it gives quite a good bit of weed control and breaks down rapidly, and that's what I needed to plant corn in that system. I didn't want too much residue in the field either. So here's mustard. It just kept growing and growing and growing. So ultimately we had to apply Roundup, and then that also didn't kill it because it was too late. Weed cover in these plots, you can see barely any weeds in these cover crop plots. There were some weeds in the bell bean plots, and that's because the residue broke down too fast. So there you can expect some weeds to come back in those plots. Other than that, other plots had barely any weeds, and you can see again there are no weeds at all in these plots. In May, we strip till planted, and I just showed that video earlier. And this is after strip tilling. We got good corn emergence. The bad part was in the plots that I had rye, I had too much biomass and it was plugging up the planter. So that's something, if you're in an agronomic system, you're worried about, but if you're in a vineyard, you're not planting anything there anyways. So not much of a concern. And here you can see that barely any weeds at all. But these are Roundup ready corn, and just as a practice, the farm manager says we had to spray. So he sprayed, but it wasn't necessary at all. We could have saved at least 1 or 2 sprays of Roundup. I don't know how many sprays he went with. I think it was 2, but that was completely unnecessary because there were fairly no weeds at all. So this is like towards the middle of the season. You can see no weeds at all. And this one was the rape plot, and it came back and it messed up the corn. And this is the standard with no cover crop, and this is with the cover crop. And you can also see that this was taken somewhere around July. Look at the amount of stress that this corn has moisture stress compared to this. So although I didn't take data, I think there was more soil moisture being held in this soil compared to this soil. And that makes sense because you're reducing evaporation here. And at the end of the season, we got good corn yields. The waviness you see is more of a factor because of the unevenness of the soil and also the irrigation system having some issues. Now, if you're thinking of reseeding, you want a self-seeding cover crop. Rye may work because we were seeing quite a fair bit of regrowth from rye, reseeding from rye. And it has the potential, but we haven't documented how much of regrowth or those sorts of things. So the question is, what is the potential of this technology in vineyards? So I'm glad that Eric and the group from Monterey Pacific shared these slides and videos with me. They're already doing this same kind of study in their vineyards, the ones that are going to be planted into vineyards. So this is from them. They've been using rye and several other cover crops. This is from their plots again. And this is actually, they have a smaller roller crimper that is going to be ideal for going in between the vine rows. So they have a roller and ring roller attached to the back. And in 1 year, I did not have my roller crimper, and I had to go with the ring roller to flatten my cover crops, and it worked perfectly. So then they will establish the vines. Here's another video showing the process again. These are all from the King City area of Monterey. So technology has already been looked at in a vineyard setting. So I'm glad to see these things happening in a vineyard setting. And that's the amount of biomass that, and so now, so that's before the planting of vineyards. So the question now is there a potential for cover crops and rolling crimping technology in established vineyards? So there must be benefits that I have seen from my corn studies in vineyard settings, but it has to be documented. So if you're thinking about roller crimper technology and you're thinking of cover crop choices, rape, Phacelia, all these mixtures are all great, except rapeseed, which didn't really break down easily or go down easily. So we needed herbicides in that case. And we get more than 70% residue till the planting of corn. In some cases, we had higher than that. That's an average. So if you're thinking about rolling and crimping, I think it's better to go late to have a good kill. If you go early, the potential of regrowth is pretty high. And you saw the benefits of weed cover, weed suppression, almost like 80%, 90% to some around that numbers. And Merced rye was the one that produced the highest biomass. And if you're thinking of ease in killing with the roller technology, Phacelia was the easiest to kill, and mustard, again, most difficult. If you're thinking of having a situation of where you don't have to, fertilize and all that, a mixture is better to go with nitrogen fixing legumes and the cereal grasses. That's probably the better choice you want to have. So I think it's time to explore the potential of this technology in established vineyards and see how they do. So with that, I'm going to end my talk and open the up for questions.
Thank you very much, Dr. Shrestha. I was just going to add one thing to the slides there in King City, in our vineyards. The cover crop that we've really isolated as most effective is Merced rye. You can get it to germinate just about any time during the year. It grows aggressively. It's tall enough when it goes to seed that you can crimp it and the stalks stay down. And the biomass is actually alleliopathic. So there's a chemical released when the plant itself breaks down that inhibits other weeds. And it just, I mean, we're dealing with a really nasty set of weeds in King City and you know, deeply rooted, sandy stuff that prefers sand, and it works great. So thank you, and please answer any questions that pop up in the chat.
So I'm looking at the questions here. How do you define conservation tillage in the 20-year cover crop study? So in that study, it was not completely no-till. Occasionally, we had to use cultivators to get rid of the weeds. Sometimes, in some years, weeds became a problem, but there was no tillage at all involved other than the occasional cultivations we had to do to get rid of some of the weeds, especially in the tomatoes, more so than the cotton. So it was close to no-till, but not quite. Next question I have is, for weed suppression, do you recommend broadcasting cover crop seeding rather than row? I think yes, the broadcasting would be, in the initial stage, yes, but once the cover crop canopy develops, I guess even the ones that are planted in rows cover the soil pretty quick and suppress weeds. So initially, the ones that are broadcast might provide more weed suppression than the ones that are seeded in rows, but in time, I think you get equal weed suppression. So I don't really have data to see if that's really true, but based on visual estimates, I think that's probably what will happen. Why crimp instead of mowing? So that's a question that, yes, we are kind of struggling with. Why crimp instead of mowing? With crimping, we seem to get a denser mat, and with mowing, again, as I said, like, if you use mowers and you select that technology year after year, you only mow. What happens is you'll select for species that are weed species that are adapted to mowing. So you will probably have grasses and some of these low creeping type of weeds that will adapt to mowing. With crimping, you're laying everything down flat and you have that residue, thick residue cover that kind of prevents weed species from adapting to that particular system. But some growers are, trying to modify. This roller crimper thing itself is being developed, so people are kind of improvising on the roller crimping technology, adding blades and all that. The next one is, how prolific self-seeding annuals like Blamdo Brome or Crimson Clover have been studied for vineyard crimping. For vineyard crimping, I'm not sure, because the only crimping study that I am aware of or saw any evidence of was from Eric's own studies there with the Monterey Pacific Vineyards. So I wouldn't be able to comment on the self-seeding, but as you saw in my study, like the rye has potential for self-seeding. So I'm thinking that Blando Brome or even Crimson Clover might reseed. The question I have in mind is, like, how late should we go so that the seeds can mature so they can reseed? So that's another question.
To that question, Dr. Shrosta, I was just going to add one thing. So with the ryegrass, with all the cover crops we've tried, we found that the taller the longer the taller the cover crop grows, longer the stalks, the more readily crimpable the cover crop. So things that are shorter stature just don't work. They pop back up. You can't get them to, you can't punch them into the soil the same way. And so you really need something that's going to get up almost to canopy height. The area where we perform those trials is, it's really cold, down to the low teens regularly. And so we were concerned about frost risk in the established blocks where we've been doing it, and we've seen no problem. We've let the cover crop go all the way until late May, till it goes to seed, so that it does come back and had no problems with frost. So I think height is key, and then there's no issue that we've seen in letting it go to seed and risk of frost.
Thank you. The next question is, have you seen any increased activities from rodents with crimping? Not really in these plots, but this is more of an agronomic system. So we, after the cover crops have been crimped, we come back with corn planters and all that. So it's more disturbed than in a vineyard setting. So what does it mean to rodent pests? I haven't seen any increase, neither in these plots or in the 5-point study that we have, that we don't really see much of rodent pests increasing. But when you move from an annual cropping system to a perennial cropping system, you'll have more vertebrate pests anyways. So will this add more to that problem? I'm not sure. Next question is, any problem with voles underneath the crimp residue? Again, I didn't see any voles. And again, I can't speak for the long term. This is 3 years, so what will it do in the long term? I'm not sure. But in the 20 years study, we didn't see much increase in voles. Next one, does anyone have roller crimper equipment in Napa Valley? Eric, probably that's a question that you can take, I think.
I'm not aware of anybody. Hopefully, it's something that people adopt and play around with. The more people who are working on it, the faster it'll be to a point that it's common practice.
Right. I had a proposal in to work with roller crimper in Vineyards. It didn't get funded primarily because I think the criticism was because I was trying it in a raisin vineyard, but that was the only cooperator I had to, see if I could work in a vineyard setting. So hopefully I will be able to work with Eric and Monterey Pacific people and take this technology further.
Absolutely.
So someone just commented there saying, yes, folks have roller crimpers in Napa Valley. The next one is, please summarize the advantages to roller crimping over mowing. Now, I didn't do a side-by-side comparison of the two to see how it differs. So maybe that's another thing to look at. So I think I will, in my next studies, I will compare mowing versus crimping to see what the benefits or bad side of one is to the other compared. So yeah, that's a good question.
Okay, well, thank you so much, Dr. Shrosta. It was really, really wonderful to have you, and we appreciate all the information you provided. If you don't mind hanging out for a few minutes, just in case there are any additional questions that pop up, you can answer those in the chat function, and we will transition to our next speaker. So next up, again, continuing with that vineyard floor theme, we have Dr. Scott Steinmus. He is a he's the head of the Horticulture and Crop Science Department at Cal Poly. He obtained his PhD, University of California, Davis, in plant biology, and joined Cal Poly in 1999, and then became the department head in 2014. He's also one of the authorities on Roundup, public perception of Roundup use, the actual risks of Roundup, if there are any, and that's a common topic these days. The lawsuit issue is something that everybody's concerned about. Roundup, glyphosate remains an important tool in the chest about every farmer in the world. And so Scott's going to discuss with us today kind of strategies for governing the social side of it, as well as maintaining the effectiveness of the product itself. And Ugo, please weigh in here. I think Dr. Steinmaß is with us to answer questions ultimately, but his presentation is going to come to us in the form of a pre-recorded video session. Correct, Ugo?
Yes. So I'll start sharing that. I don't know if Dr. Scott, you want to say a couple words before we project the.
Oh no, that's fine, Hugo. But that's right. This talk is going to be about resistance from everything, from people to weeds, and so that's what we'll be talking about. I'm borrowing a little bit from my California Weed Science Society talk that I gave last January, but then I've also added some other stuff about weed resistance to especially glyphosate. But go ahead, Hugo.
Got it.
Hold on, Hugo. The sound is.
I'm sorry.
Not coming.
Was there a problem?
We're missing the volume.
The sound, yeah.
Give me 1 second. Give me 1 sec. Just really quick, was there any audio on it?
Yeah, there was.
Okay, I'll play it then.
If you go to your microphone, I think if you select same as system.
But you are able to hear it now, right?
I can hear you.
Let me see. So I changed it.
There's sound on the video or something else you're asking, Scott?
There's sound, yeah, there's sound in the video.
Oh, there's if you go to view options, I think there's something there that says share sound from laptop.
Okay, give me 1 sec. See if that
Hey, if worse comes to worse, I can.
I'm going to be talking about vineyard weed management on the vineyard floor. And in particular, I'm going to be talking about the resistance to herbicides that people and weeds have.
Thanks, Ugo.
And specifically, I'm going to focus this talk quite a bit on glyphosate. This chart here comes from the California Department of Pesticide Regulation. And what it shows is it's the latest reported pesticides that have been applied in California. And these are the top 5. What you notice both in pounds of pesticide applied and then also the cumulative acres, you notice that glyphosate appears in both these charts. And so glyphosate is a major pesticide that's being applied in California. This slide here shows you the past several years up to the latest reported year of 2017 of the top herbicides that are applied in grape-bearing vineyards in California. And you notice here that glyphosate tops the chart. And you'll see the active ingredients on the right-hand side in the table, and you'll see the brand names that many of you will recognize. So how glyphosate fits into this picture of pesticides being used comes down to Proposition 65. And what Proposition 65 is, it's regulated by the California EPA, and according to the Safe Drinking Water and Toxic Enforcement Act, it gives the governor the duty to report and republish every year chemicals that are known carcinogens or reproductive toxicity factors. And so the California EPA, what they did when they do this assessment for glyphosate, they use IARC's assessment. And what's important here is that IARC, International Agency for Research on Cancer, which is a group that is part of the World Health Organization. IARC represents something of you could consider a global EPA, and so it's a very important body. And what they did was they did what's called a hazard assessment. And what's different about hazard assessment and risk assessment is that hazard is really just establishing a threshold of a chemical that could cause cancer. So can a chemical cause cancer? Whereas a true risk assessment combines that threshold, that concentration, with the likely exposure, and puts those two things together. So this is the list actually is 23 pages long, and I'm just showing you a few of the compounds that are on the Prop 65 list. And of course, glyphosate. You can see glyphosate shows up down here in the toward the bottom part of this list. And I'm not trying to make fun of the kinds of things that get included on this, but you can see how very common everyday compounds and particles are on this Prop 65 list. And you'll see that, again, what's really important here is that IARC is what was used to assess glyphosate hazard. And it was a hazard assessment and not a risk assessment. The U.S. EPA, what they are a regulatory body that does risk assessment, where they combine the hazard with the probable likely exposure levels to come up with an overall risk. So what's happened because of this, possibly as a consequence of this, it's probably very complicated, is that consumer groups are starting to fight back against glyphosate. And so you see here, this is just one example, the Detox Project that you'll see and usually they're pretty high-end stores where you'll see this, you'll see these symbols about glyphosate residue-free produce. But you're also seeing this in the wine industry. So here's an example with Smart Wine. They're part of this Detox Project with this label of a glyphosate residue-free. And so consumers are starting to demand wines that are free of glyphosate, and that essentially, whether glyphosate is there or not, they want vineyard managers to not use glyphosate. So as a consequence of that, the wine industry has fought back. And so this is an example from the Wine Institute, a clipping from their website that talks about glyphosate and its overall risk. And what I like about this, about what the Wine Institute has done, is that they're taking regulatory bodies like the U.S. EPA. They're also using the European Food Safety Authority and then other regulatory bodies, right? So it's not just a hazard assessment that they're looking at here. They're looking at the risk assessment, which is the exposure. It's the hazard, which is what's the concentration that can cause cancer, combined with the likely exposure levels for a particular group. And so here's a little bit of a quote to put it in perspective. And so what Professor Winter did here from UC Davis is he's basically saying that a person would need to consume 140 glasses of wine a day, every day for 70 years, and you would still be under the level of glyphosate considered a no significant risk. And what Dr. Winters did was he used that Cal EPA hazard threshold of 51.4 parts per billion. And so what Dr. Winters is doing here is really he's taken that hazard from Cal EPA and putting it together with the likely exposure level to come up with an overall risk. And you can see here that 100 risk. So does glyphosate cause cancer? This is a very, very complicated question and very difficult to answer. And the reason why it's so difficult to answer is because when you're considering glyphosate carcinogenicity, from a statistics standpoint, you're taking pieces of information that make it very, very complicated stats problem. So, for example, we're talking about low exposure rates. In the relative sense, the population that we're considering, we're considering very low exposure levels. So also long duration and lag periods between the exposure and when the actual consequence of something that you care about, like cancer, when those are long, then it becomes very difficult statistically to detect these effects. Also, rare events, for example, non-Hodgkin's lymphoma is the main target for the cancer that glyphosate is being implicated in. So when you have low exposures, long durations and lags, and then rare events, those things are very, very difficult effects for statistics to detect with any certainty. So also there's some implications that glyphosate and certainly other herbicides might be endocrine disruptors, and so those effects usually show up in the next generation. So that just adds another level of complication that statistics has a very difficult time detecting. So the other part of it is confounded effects. So what formulations of glyphosate are you talking about? You know, and there's so many other factors that interact and confound the data that comes in from these studies. For example, does a person smoke or not? How old is the person? What's the occupational regional exposures to other things that can cause the same cancer? So as a consequence, it's a very difficult, taking these confounded effects into account makes it even more difficult for statistics to determine a true effect. So then there's also bias. So for example, information bias. Information bias might be something like, what was the true exposure level for a person? And so if you're going to try to assess risk, did you get the exposure level right? The selection bias, like who gets picked for these studies? Is it people who just have cancer, or is it just a random selection of population? Recall bias, for example, people who, this is very common bias, where somebody who does have cancer and they've read about these kinds of cancers and glyphosate links, their recall tends to be that they were exposed to glyphosate, whereas really they weren't. And so this is or not at the level that they think they were. So and then there's also proxies. For example, when agricultural workers, if they're not reporting to the data collecting agency, they have other people do it. So proxies usually don't get things exactly right. So these are biases that can make statistical detection of these effects extremely difficult. Then there's also just chance, random variation, you know, and then exposure misclassification. That's a form of information bias. Like, did somebody really get exposed to glyphosate? For example, a worker in a Roundup Ready soybean field in Iowa, they may be in an enclosed cab where glyphosate is being sprayed. That enclosed cab could have a HEPA filter. So the person who's had that tractor spraying glyphosate could be exposed to air that's cleaner than any air that we have, where we don't spray glyphosate, right? But the person was around glyphosate, and so that could be a form of misclassification. So these could be important and they may not be important. So this is just all of these things together just make this whole does glyphosate cause cancer makes it an extremely difficult question to answer.
Hello there.
So what's important when you're trying to answer these types of questions is to basically quantify or try to quantify a type 1 and a type 2 error. So in statistics, we have these two types of errors. Let's walk our way through this. What we're going to look at here is we're going to look at two populations, right? They basically represent they represent the people that were selected from the same group of people, but one population of selected people are going to not be exposed to our carcinogen, and the other group the other population that we've selected randomly will be exposed to our putative carcinogen. So we have two groups here. So in our first group, if the average cancer prevalence for that particular group that was not exposed to a putative chemical that causes cancer, its average cancer prevalence might fall with this U0, or this right here. So what we do in statistics is we usually set a threshold, right? And so this is represented by this asterisk right here. And the asterisk, that threshold is usually set at 5%. You know, so sometimes we go 10%, sometimes we go 1%, but usually, very often, most of you probably learned this in college classes, that 5% are our significance level. And so what that represents is 5% of the population that might get cancer, but they weren't exposed to the carcinogen, right? So it's just chance that they ended up getting cancer. So what we want to know is, when we do expose the population to glyphosate, we want to know what their average cancer prevalence is. And if their average cancer prevalence rate turns out to be higher than that asterisk, then as a statistician, you're going to conclude, well, then this stuff causes cancer. So that's what the significance level is. But we still, what could happen if we don't know what the true distribution, that red curve right there, we very often don't know that red curve. And so basically, when we do our study, if we end up with an average that's above that asterisk level, we could still be wrong by 5% of the time, right? Because the person in that group, that 5%, could have gotten cancer just by chance alone. But if we do have the true distribution, that red curve that you see over here on the right-hand side, then we can actually come up with what the true type 1 error rate is, and then we can also come up with what's called the type 2 error. So the type 1 error is basically it's somebody from the population that was not exposed to a putative carcinogen, but they ended up getting cancer, right? So that's a false positive. That's a type 1 error, a false positive. When we do know the curve, the true exposure curve for this red curve on the right-hand side, we can also calculate what's called the type 2 error. What the type 2 error is basically it's a false negative. So if we end up doing our studies and we end up with an average cancer prevalence rate of an exposed group that is on the left-hand side of that asterisk, our threshold value, right? Then that's a false negative, because you could by chance select people who were exposed that might not have gotten cancer and conclude that it doesn't cause cancer. That's a type 2 error. That's a false negative. So type 1 error is a false positive, and type 2 error is a false negative. So that's what we're trying to get at, right? And so from these it's actually 1 minus the type 2 error is the power of that test. And so the smaller that yellow shaded area is, you will find the power of that detecting that effect. Does that chemical cause cancer? That power goes up. So that's what the power of the test is, the area that's over here on the right-hand side of the yellow. So how does that pertain to everyday information that we use when we try to assess and set standards for whether something causes cancer or not? Well, we use what's called a dose-response curve. And what a dose-response curve is basically the response is going to be the event, which is what percentage of the population get cancer. The dose, of course, starts at very low doses, next to 0, and then goes up to very high doses. And so what we want to do is establish this dose-response curve to a particular population that we care about. Of course, we can't do this with human populations because that would be unethical. So we try to do this with rats, or we try to do it with other organisms, right, where we try to develop these dose-response curves. But the problem, even with the dose-response curve, is this. All right, so the best number that you can get off of a dose-response curve is the median value. What dose causes 50% of a population to get cancer, or whatever event that you're interested in, right? And the reason why that's so important is because you'll see that the confidence intervals, that bar that you see right there on the red line, that confidence interval is the smallest for a dose-response curve at the median. And so it's the most precise prediction, and as a consequence, public policy is based on a median value or some factor of a median value. Now, what you'll notice here is that you'll see I'm looking at low doses and high doses, and you can see what the confidence intervals look like, right? They're actually rather large. And so what ends up happening, this relates to the bias, right? So people who do not rely on the chemical as part of their livelihood, for example, the general public doesn't really rely on glyphosate as a tool for their livelihood. And so what they want to do is they want to control for the type 2 error. They don't want to commit a false negative, right? But the problem is that they tend to use stats, and they tend to have biases that are prone to a type 1 error, a false positive, right? Saying that it causes cancer, but it doesn't really cause cancer. On the other hand, at the other end of the curve, people who do rely on a chemical like glyphosate as a tool for their livelihood, they try to control for the false positive rate. They try to control for the type 1 error, right? Problem is that they're prone to the opposite error on the other side of the curve. They're prone to a type 2 error, right? They're prone to a false negative. So you can see that the motivations here are completely opposite depending on whether you depend on the tool or don't depend on the tool. And you know that 99-point-something% of the general population does not rely on glyphosate as a tool for their livelihood. So they can afford to say, let's ban it. So the most robust studies to date. So this is really I'm summarizing a long talk that I gave in the California Weed Science Society last year. And so this is basically just a summary of the studies. And so there are details that I can relay to everybody in my slide presentation as well if you want to contact me later on. But the most robust studies to date. So IARC, the European Food Safety Authority, the U.S. Environmental Protection Agency, the reason why they come up with different results is because they use different studies. And they focused on slightly different questions. Again, IARC is a hazard assessment body, right? They just want to know if this causes cancer, like Cal EPA. Whereas the European Food Safety Authority and the US EPA, they're regulatory bodies. And so they do risk assessment. So they take a hazard and they combine it with the likely exposure levels. So it's a different question. So that's why they come up with different conclusions. So this makes it really confusing for the general public. And if you don't rely on the tool, it's going to be easy for the general public to say, nope, just ban it. So the most robust, some of the most robust studies, because we can't do dose response curves on human populations, we need to rely on case control studies or cohort studies. And so the best studies out there right now is this prospective cohort study called the Agricultural Health Study. It's the longest ranging, the most intensive investigation of people exposed to glyphosate and other pesticides in the farming environment. This has been going on since 1993, and so no AHS study has found a positive association at typical farm exposures for glyphosate and the link to cancer. So probably the best study, you can argue this point if you'd like, but probably the single best study out there is this prospective cohort study that's based on the AHS data set. And it's from 2018, Andriotti, DeRusse, and there's a bunch of other names that are big in this topic. So what they did was, even with the highest intensity weighted lifetime exposure levels to glyphosate, they found a relative risk of 1.12. That's basically saying a 12% increase in the risk of cancer. But look at the confidence intervals. When the confidence intervals span 1.0, 1.0 is background level, which basically is equivalent to saying no effect. So you can see here that the significance level is not significant. And this is after a 20-year lag after this population were exposed to glyphosate. Meta-analyses they combine a bunch of studies that have looked at the same question and try to come up with an overall risk, right? So probably the best meta-analysis that's out there on glyphosate is probably the Zhang et al. 2019 meta-analysis. And what they did was they came up with a meta-relative risk of 1.41, which is equivalent to saying 41% increase in relative risk of non-Hodgkin's lymphoma. And unfortunately, that confidence interval did not span 1.0, so it's a significant result. But what's noteworthy here is that all the studies were case control studies, case control studies can be not quite as powerful as cohort studies like the Andriotti study, but the Andriotti study was included in the Zhang meta-analysis. So again, I'm sorry, it's confusing, but this is where we're at. What's really important is that you can't establish causality based on one study. And so that's a really important point. So the summary of the issues that remain with glyphosate and whether it causes cancer or not. So there's been lots of criticisms, lots of detailed criticisms that you have to be a statistician or an epidemiologist to really understand if those criticisms are legitimate or not. But one of the things was that they used imputation to fill in missing data. And so there's a criticism of that imputation method. Well, so Andreotti, like good statisticians do, they basically say, well, let's do without the imputation method and see what happens. Well, no significant effect still. So that's what you do, right? You try to, so with a meta-analysis, you take a study out, see what happens to the result, put a study, and you do it one at a time. And you try all these different combinations to see if you get a different story. When you do, then you have to really start digging in. All right, so the Andreotti study still holds up pretty well with some of the criticisms that have come up. For the weighted studies, like a meta-analyses that have been done, these are really complicated nuances, like do you treat factors as if they're fixed effects or random effects? For example, if you treat something as a fixed effect, unfortunately what ends up happening in the statistics is you end up with artificially narrow confidence intervals. If you think a little bit about artificially narrow confidence intervals, you will realize that you're increasing the type 1 error. You're increasing the chance of committing a false positive, saying that something causes cancer, but it does not really cause cancer. So whether something's a fixed effect, random effect, the best way to do it is to try both. And then also the meta-analyses really suffer from this quality assessment, and the fact that they don't use quality assessment to weight the studies. What they do is they use the standard error from each of the studies as the 1 over the standard error as a measure of how strong that study is as far as coming up with its conclusion. But why not use the quality assessment, right? Like how well was that study conducted from a statistics standpoint, not just the standard error, because you can get these standard errors pretty small if you have really high sample sizes. So it's still complicated, very complicated. And so the other really big question is what is the true exposure level? And I gave you an example. Somebody who has who might appear to have a really high exposure level, like a Roundup Ready farmer, their highest exposure level could be climbing out of their enclosed cab, their HEPA filter enclosed cab, after spraying an entire day Roundup on a Roundup Ready soybean field. They climb into their pickup truck, and then they take off all their PPE, and then they realize halfway home, oh, I forgot my cell phone in my back of my enclosed cab. They drive back, they don't have a glove on, they grab the door handle from the outside. That could be their true highest exposure level, which is basically nothing. Whereas a homeowner, right, who may not know or may not respect PPE, personal protective equipment, may not be wearing gloves, may be spraying Roundup from a squirt bottle, right? And it's leaking all over their hands. And so it's really hard to tell what the true exposure levels are. And then exposure level to what, right? Different formulations of glyphosate exist in different countries. And so what are they being exposed to? Is it really the glyphosate, or is it the surfactant that's maybe augmenting the problem? So we don't really know. So let's go back to resistance from a biological standpoint. So let's go back to what we actually study, right, which is weed science. So as far as fighting back, right, so the public has fought back against glyphosate. Well, turns out that weeds also fight back against glyphosate. Since glyphosate is the #1 used herbicide, pesticide, and often pesticide in vineyards, of course the weeds have fought back, right? So these are the known cases of resistance in weed populations. And what we see here is we see that EPSP synthase, which is glyphosate, which is the WSSA code G for mechanisms of action. You see here we got horseweed, jungle rice, hairy fleabane, and then Italian ryegrass. Italian ryegrass is your most likely candidate to be resistant to almost anything. And so you'll see here ryegrass has also got multiple resistance, which means that you have 3 different, very metabolically different pathways of resistance to these different mechanisms of action. To a weed scientist, that's extremely scary. So there's Italian ryegrass right there, and then there's horseweed, also known as mare's tail. So there are 2 general categories of herbicide resistances out there. The first one is target site resistance. And so the 2 subgroups of target site resistance is increased expression of the target protein. So for example, EPSP synthase just might get overexpressed in a tolerant plant. And so when you spray glyphosate, there's not enough glyphosate to bind all the protein, the EPSP protein that's being expressed. And so the plant can actually live. It can actually, the EPSP synthase that didn't get bound by glyphosate can do its job. The other is something that you may have heard of, and this is common, is that the target site has a mutation in it, so that the herbicide can't bind to it. This is a very common one. This is definitely one that's happening in ryegrass. So the second big category of herbicide resistance is called non-target site resistance. And this is how this works. Basically, you can have reduced penetration of the herbicide. So for example, thicker cuticles or hairs on the leaves will prevent herbicide from getting into the plant, right? So that's reduced penetration. Thicker cuticles might provide this non-target site resistance. There can also be altered translocation and compartmentalization. Many of you already know about this, right? Like with bindweed. Field bindweed is really good at compartmentalizing herbicide, so it doesn't move very far in bindweed. So that's why you don't get a very good kill with field bindweed, or something like Johnson grass is also very good at compartmentalizing. So enhanced herbicide metabolism. So all plant cells have in their endoplasmic reticulum, they have these cytochrome P450 monoxygenases that are generalized oxidizing enzymes that can break down many toxic compounds from the outside environment, including herbicides. Certain plants are better at producing these than others, and so they become resistant to herbicides, including glyphosate. So there's also enhanced neutralization and protection. So for example, paraquat, the mechanism of action for paraquat produces a free radical that destroys plasma membranes. Some plants have oxidases and peroxidases that are really good at neutralizing those free radicals, and so the plant can become tolerant of paraquat because of that, because of these oxidases and peroxidases. So these are the two general categories I want you to pay attention to: target site and non-target site. So as much as target site is scary, it's non-target site that really concerns us the most, and the reason is because these generalizable resistances with non-target site is what's often responsible for multiple resistance. When you have resistance in a single weed to many different, very, very different mechanisms of action for herbicides. This is a diagram right here, and we'll make these slides available for you. So if you want to take a look at this later, this slide right here basically just shows you the different what's supposed to happen. So up here at the very top, this line right here shows you what's supposed to happen with a herbicide. The herbicide is the red mushroom shape symbol that you see right there. What's supposed to happen is supposed to penetrate the leaf or whatever target that you're talking about. It's supposed to translocate to its target site. And so, with EPSP synthase, it's wherever amino acids are needed. And so the herbicide will bind to the target with glyphosate. It is EPSP synthase is the target. And once glyphosate binds to the target, then the substrate, PEP, cannot bind. And so what ends up happening is the plant will die, because in this case, it runs out of amino acids. Okay, the two forms of herbicide resistance, target site and non-target site, is down here in this next row right here. So you see here, so reduced penetration is an example of non-target site resistance. Reduced translocation and compartmentalizing, that's an example of non-target site resistance. Some plants are better at others than that, which leads to their resistance. Overproduction of the target protein, right? So that's an example of target site resistance, remember? And so then also the mutation that we see right here, the mutation in the target protein so that the herbicide cannot bind. For example, this FC category right here, that's what the EPSP synthase mutations in ryegrass is. It's basically complete tolerance of glyphosate. The glyphosate cannot bind at all, but the substrate can, so the plant lives. So enhanced herbicide metabolism, that's a non-target site resistance up there. And then we also have peroxidase activity or some oxidases that can neutralize free radicals. So with EPSP synthase, that's not the case that we're talking about. Something like paraquat or a PPO inhibitor like goaltender, herbicides that are plant weeds that are resistant to those mechanisms might have this neutralizing mechanism, which is a non-target site resistance. So how do you know if you have target site resistance or non-target site resistance in your weed populations? Well, what you do is you're going to look at this. You're going to look at this. You're going to look at herbicide rate, and with herbicide rate, if the population is when you spray it as you spray it at an increasing rate, you'll see that a susceptible biotype of your weeds will basically die as the rate goes up and up and up, right? And so you'll get to a certain point where you have some median tolerance to that particular herbicide, and then you might have the highest labeled rate, which is getting almost every single susceptible biotype. But if you don't get every single susceptible biotype, you can start to see some developments of resistance. Let's take a look and see how that would look for target site versus non-target site. So if you spray at this rate right here, all right, so you're not spraying at the highest labeled rate, you're going to spray at some other recommended rate because you're trying to save money, and you've gotten pretty decent control. You're going to have a little bit of survivors, right? Because you'll see right there, those are all survivors. So if of those survivors, if you see this kind of dose response, right, with those survivors, where basically you have to spray at a super high rate in order to get those last few, those individuals that survive that application right there, that's a pretty good indication that you've got yourself target site resistance. It happens very quickly, and you'll see it very distinctly, right? It'll be very obvious. You know, you'll go from a 5% weed population to a 60% weed population in a single year if you're experiencing target site resistance. On the other hand, you might see a slow development of resistance, right? So where the survivors, basically you can still spray at a higher rate and get many of those survivors. But if some of those, basically what will happen with non-target site resistance is you'll get this slow shift to the right. And that slow shift to the right of tolerance is a pretty decent indication that you're dealing with non-target site resistance. And so it takes a while for that to happen. And so that's usually the two indicators that help you identify which one is happening. So studies have shown that spraying at low rates tends to lead to non-target site resistance. These examples exist in glyphosate, ACC inhibitors like the DIMs and the FOPs, like cefoxidim, or fatty acid inhibitors. And then there's also, but by the same token, spraying at high rates, you're going to see target site resistance appear even more quickly than if you spray at low rates. And so there's no real true solution to this problem. Identifying whether it's target site or non-target site is extremely important. If it's target site, you got to switch mechanisms of action completely. If it's non-target site, you may be able to spray at a higher rate and still get all those survivors. Okay, let's go back to those mechanisms of action in a plant cell. So I'm showing you here, these letters right here correspond to the different mechanisms of action codes. It's part of the Weed Science Society of America and the Herbicide Resistance Action Committee. And so you're going to see some brand names here that fall into each of these categories. So for example, Chateau falls into this E category of PPO inhibitors. Rely, it's a glutamine synthase inhibitor, falls into this H group right here. Roundup also falls into this H group because it's an amino acid inhibitor. You'll see that Goaltender falls in is another PPO inhibitor. You'll see that Gramoxone is a photosynthesis electron diverter, which also falls into the CDE category. It's a D category of herbicide. You'll see over here Prowl. This is one of the LENs, tubulin inhibitor. This also falls into group K. Allion, which is indazoflam, is a cellulose inhibitor, falls into this L group of herbicide mechanisms of action. You'll see Surflan is also a LEN, and so it's a tubulin inhibitor, so it's a cell division inhibitor. It's very similar to Prowl. So 4 ways that weeds are resistant to glyphosate. The 4 ways that weeds are resistant to glyphosate, basically target site resistance, so it's an altered binding site, so it's a mutation. We already talked about that, and that's very common. So it's position 102 or position 106 on the EPSP synthase gene, amino acid that makes up that protein. So those are, and that's basically all or nothing resistance, which when there is a mutation, it's all resistance. Its highest rate of glyphosate can't take it out. Non-target site resistance is also been found with glyphosate. Also, the production of the breakdown enzyme, GOX, right? So GOX is glyphosate oxidoreductase, and that's at P450, generalizable oxidase. Then there's another form of non-target site resistance, altered uptake and translocation of glyphosate, have been found in biotypes resistant to glyphosate. So not all of these exist in grapes for weeds that grow in grapes, but they exist in other cropping systems, and it's probably a matter of time before these also appear in grape weeds. Okay, let's begin the healing. So what is the solution to the problem of weeds developing resistance to herbicides, especially glyphosate? What's the problem with people developing resistance to herbicides, especially glyphosate? Well, the answer to both those problems is integrated weed management, a component to integrated pest management. And so, as you remember, integrated pest management is basically utilizing methods that provide acceptable weed control that begin with the least impacting from a social, environmental, and economic standpoint, then adopting more impacting methods as those previous methods don't work. So first component to an integrated weed management program is to understand your crop and its timings. When do the different operations happen? When do weeds come out? What is the crop doing? Then knowing your weeds, weed identification, super important. Monitoring weeds at times that are appropriate. It doesn't make any sense to monitor for weeds in July because most of the time your weed problem is already done by then, and it's not raining. And unless you irrigate all over the vineyard, weeds are only going to grow where the water is growing. So appropriate timing for winter weeds, which is in November after the first rains, and then also summer weeds, which will start to appear in February or March. So the methods that actually kill weeds are these. These start from the least impacting, then go into the more impacting. So prevention, cultural, physical, biological control, and then chemical control. We don't have very good biological control mechanisms for weeds. They're very good for insects, and biological control is very effective in wildlands. But in cultivated agriculture, including vineyards, we don't have very good biological control yet. This is showing you the different timing of operations in California vineyards. You'll see the different months of the year on the far left-hand side, and then you'll see the different operations that go on inside of a vineyard. I haven't included them all. I haven't included pruning or flowering or any of those, but many of you know how those fit in with the timings that I have included. And then we're talking about irrigation when water is available. When we talk about drip irrigation, just remember that drip irrigation is often only in the planting line right next to the vine. And so wherever water is with exposed soil is where weeds will start to grow. Then the next column over, you'll see over here, these are the different possible operations you might have for herbicides. So contact herbicides soon after harvest. You might have some pre-emergent herbicides just as maybe before the summer annuals start popping up. Winter annuals might be around, so you might have some post-emergent herbicides for winter annuals. And so these just show you some of the different herbicides that could be used. You'll notice here that I've included glyphosate with a big question mark right here, and you'll notice that I put that after bud break. If you want to minimize any chance of glyphosate ending up in wine grapes, which is highly unlikely no matter what you do, because if glyphosate is going to end up in wine grapes, it's going to kill your vine first. So this is often not a problem. But if you really want to minimize even that perceived risk, just don't spray glyphosate after bud break. So these show you the different annuals, winter annuals up here, and then summer annuals when they might pop out. So from an integrated weed management standpoint, one of our first things that we need to do is identify our weeds. For most vineyard managers, after the first year or two, you pretty much know what your weeds are. The weeds that keep you up at night are usually about 4 to 6 different weeds that might keep you up at night, and you learn them in the first year. And so usually you don't need these identification tools after the first year. But these are some of the tools I use with my students, and what's important with these tools, especially if you're using an app like this, is to take many, many pictures, many different stages of the same species, same plant, so that these tools have the best chance of identifying your plant. And then verifying your weeds with the University of California Integrated Pest Management Program. On their website, they have a weed gallery that has really good pictures that you can help verify your weeds. So I don't have time to talk about all the possible methods you could be using. I'm just going to give you a few of them. And so we're going to start with some of the mechanical physical methods that might be utilized. So many of you have seen some of these kinds of rototillers and French plows, right? These are mechanisms. They often have these mechanical trips that will help the plow or the tiller get around the grapevine if you're trying to take out weeds in the planting line. The middles are relatively easy. You can use a disc or you can use a rotavator. So many of you have seen these. You can also use these other physical methods like flaming or steaming. These can be very effective tools, but they're very effective only on small weeds. So for example, this weed right here, this weed was flamed, and you can see that it looks like it's wilting and dying, but it won't die. That plant will easily recover from flaming. So it's important to get these when they're really small. These are cover crops that we've used in the past. This is from a past experiment. So what I'm showing you here is a cover crop of oats and vetch being grown in the middles between the vine rows. The vine row is right here, and then the middles are right here. So we're growing a cover crop in the middles right there. And then we're coming in springtime, April or so, and we have this special mower that will take out the cover crop in the middles, and then it will blow that mulch into the vine row right here, and it will pile it up to suppress weeds. And so doing this on a 3-to-1 ratio, where the cover crop's grown in a factor of 3 and then piled up into a factor of 1, you can pile up that mulch very deeply, and it will suppress weeds very effectively. And you'll get some residual control into the following year as well. So it's really good at controlling broadleaf species. There are other types of mulches out there like weed cloths, and you also see wood chips can also be used. And so these are also tools of basically suppressing light so that weeds can't grow. But what I want you to notice here is that there's field bindweed right there. Very common weed for vineyards in California. So this is showing you that grasses are pretty good at getting through mulches because they have this life form where the coleoptile comes up like a spear and just weaves its way through the mulch and can come up. So mulches don't do a great job of controlling grasses. And also, in some of our studies, we found, many of you know John Roncaroni. He and I were involved in this project that I just referenced in the previous slide. We found bindweed was basically everywhere. So we had this quote that we said, well, there's 2 kinds of vineyard growers out there, those with field bindweed and those that will have field bindweed. And we consider that, Clyde Elmore has considered this probably the most difficult perennial to control in California. Also, another problem with using cover crops can be some of these vertebrate pests, like voles or some possibly gophers. And then also another problem with cover crops is that the green ground cover can increase risk of frost, but only during that period of bud break when it's susceptible. So these are some of the risks that come with cover crops. Now back to the different herbicides that are used in grape-bearing vines. These are the top 5 active ingredients. You've already seen these, right? So there's Roundup, there's Rely, Goaltender, and Dazaflam or Allion, and also Matrix, right? So I'm giving you some examples here, target site and non-target site resistance for each of these mechanisms of action. So they exist, right? And so for some of these, they don't exist yet. For example, in Dazaflam, there are no known resistance cases for the cellulose biosynthesis inhibitor in Dazaflam, the L29 group, but there is for the L21 group. So, which is another group of cellulose inhibitors. And so it's just a matter of time before we start seeing resistance to Indazaflam. With Matrix, which is rimsulfuron, sulfonylureas, many cases of resistance to ALS inhibitors. And so you need to be very careful when using this over and over and over again. Notice here in these top 5 active ingredients, notice that from the previous assessment of the top 5 active ingredients in vineyards, Chateau and Gramoxone fell off this list for this current list. 2017 is the latest one reported. And so Indazaflam, Allion, and Matrix took the place of Chateau and Gramoxone. And for Gramoxone, it's probably good that that's dropped off the top 5 because of its toxicity. So as far as the top 5 herbicides used in grapes, I'm giving you examples right here. Again, I'll make these slides available to you so you can see what we've got, you can see for yourself in detail what's going on here, the non-target site and target site resistances. We see several weeds in grapes that are with glyphosate. And then Rely is glufosinate, glutamine synthase inhibitor. So is there non-target site resistance? Maybe. So we see this in ryegrass with grapes. Oxyfluorphan is a goaltender, is a PPO inhibitor, so target site cross resistance with other E group herbicides, the PPO inhibitors, like Shark, which is carfentrazone. So being careful with that and paying very close attention to those mechanisms of action. And Dazaflam so far has been there are no known cases of resistance, right? But there are other L group herbicides that do have resistance, and so it's just a matter of time for Allion. But so far we haven't seen anything with that one. And then in Matrix, there are many possible mutations that can give target site resistance to the ALS inhibitors. So you need to be very careful with these. What's good about this group of these top 5 herbicides, they're very different mechanisms of action. That's what's very good. What's most likely to come back if glyphosate gets banned is probably Chateau and possibly maybe a contact broad spectrum herbicide, something like Shark or carfentrazone might take the role of glyphosate if it gets banned. This is just showing you the whole list of other herbicides that are registered in grapes, and what I want you to pay attention to is these red boxes are the different herbicides within a mechanism of action. So if you do rotate, you want to make sure you get herbicides that are outside these red boxes. Don't recommend herbicides within a red box. So you see the tubulin inhibitors, the PPO inhibitors, and then the DIMs and FOPs like fusilade and post. So staying outside that. The problem with a lot of these herbicides is that many of these have issues, quote unquote. So toxicity like paraquat, right? Extremely toxic pesticide, or groundwater protection like simazine is a known groundwater contaminant. So then there's also application restrictions, like even with Dole, right? You have that February date for applications. So the OMRI certified organic herbicides, these I would be remiss to miss out on to not mention these. These are some that are out there: clove oils and the cinnamon oil. We also, so there's different formulations of the clove oils. These work really well on small weeds. It's a lot like flaming. It's a lot like using a steamer or a flamer. You have to get the weeds very small. It's usually burn down contact, and so you have to get them early, or these just will not work. Many of the vineyard managers know the combinations that work best for them, and I'm just showing you a couple common combinations out there, showing you this excellent weed control. Right? And you can see the bare ground where the herbicides were actually applied, and you see these very common combinations. They're tank mixed, like Allion and Chateau, or Goaltender and Roundup. So you see these very effective, extremely effective. If you keep doing this before they set seeds, before weeds set seeds, you can actually ride that gravy train for a while, because if no weeds blow in from the outside, like bristly ox tongue, then you will experience some pretty decent residual benefit. So resistance management strategies. So as I finish up this talk, what's really important is to use different modes of action, and specifically different mechanisms of action. A mode of action is more general, and if you can rotate modes, then that's great. That's way better than rotating mechanisms within the same mode. For example, photosynthetic inhibitors. There are many mechanisms of action that are photosynthetic inhibitors, but if you can do a photosynthetic inhibitor and then also an amino acid inhibitor, that's a really different rotation. So from year to year, if you use different modes of action, that's pretty good. That's called rotation. You can also do mixtures in the same season, where you have different mechanisms of action in the same season. And so what ends up happening is that mixtures tend to be better than rotations. So if you have two or more different herbicides in the same tank on the same season, it's better than rotating from season to season with different each season. And the reason is because the resistant biotype of weed usually doesn't suffer that much from a competitive standpoint when you don't use the herbicide. And so the susceptible biotypes can't outcompete them. And that's what you bank on when you use rotations. And if that's not true, if there's not a big fitness cost for the resistant biotype when you don't use the herbicide, then rotating doesn't work very well. So that's why mixtures are better. It's important that mixtures that you not only they have to not only be different mechanisms of action, but they need to be very different metabolic pathways. And I just mentioned that. So a photosynthetic inhibitor with an amino acid inhibitor is a very different metabolic pathway. Very difficult for weeds to develop resistance to that kind of mixture. Also, we don't fully understand non-target site resistance, and so there's going to be a lot of studies that we need to really, really fully understand that, but they are very insidious. These forms of resistance are very insidious, and they lead to multiple resistance, which very much scares us. It's important that when you have mixtures that the herbicides have the same level of efficacy. Because what happens is if you have a herbicide that has 90% efficient efficacy mixed with a 50% efficacy herbicide, the weeds will develop resistance much faster to the 90% efficacy herbicide because that's a higher selection pressure. So you tend to find resistance. So it's important that you have the same efficacy level for the herbicides that you tank mix. So okay, some more examples of resistance management strategies that work. What's important is to not, it's not efficient to mix in a herbicide to which a weed might be resistant to one of them. So for example, if you've got Roundup resistant horseweed or mare's tail in your vineyard, it doesn't make sense that you're going to tank mix Roundup with some other herbicide, because all you're doing is providing a selection pressure for horseweed to develop resistance to that other herbicide. It doesn't even see the Roundup. So it's better to have a completely different herbicide than Roundup that the mare's tail and horseweed might be resistant to. Spraying herbicides down the planting line and then using some kind of mechanical method down the middles might be efficient, right? Because what you're doing is you're keeping the susceptibility alive in the genomics of the weeds in your field. And so by keeping susceptibility alive in your vineyard is a way to keep that herbicide working in the future. And so this is very similar to the refuge concept that's used in Bacillus thuringiensis crops in the Midwest, and it works. And so this is using different methods in the same vineyard can be very, very effective at preserving the efficacy of your herbicide. Integrated pest management using combination of prevention, cultural, physical, chemical methods, super important and can't be overemphasized. What's important is to prevent seed production, because what seeds represent, once it gets into the seed bank, is a memory of past management practices. How good of a weed manager were you? And then a momentum for the future. What will you be dealing with in the future? So a seed bank is your enemy. This is the summary for this talk. So basically, resistance to herbicides by both people and weeds is a very complicated problem. And we're seeing that glyphosate is a major target for both people and for weeds. Glyphosate is a major target that, because it's the #1 used herbicide in vineyards, this is a serious problem. For weed resistance, you need to understand target site and non-target site resistance. We tend to see target site resistance develop fast. We tend to see non-target site resistance develop slowly, but it's the non-target site resistance that scares us the most because it often leads to multiple resistance. And again, like for example, Allion and Dazaflam, we don't know any cases of resistance yet, but there are other L groups that could kind of contribute to non-target site resistance to that herbicide. For people, this is a very difficult problem, right? And I don't know how to answer this problem. Understanding statistics usually is not a solution for the general public, from what I've discovered. And so this is a very serious problem. Integrated weed management really is the answer to all, and that's what people want to see, is that essentially you're going to use herbicides as a last resort or as a last tool to control weeds. Incorporating resistance management strategies will satisfy all, and both from a weed resistance standpoint and from a people resistance standpoint. And so with that, I'll take any questions, and thank you.
Thank you very much, Dr. Steinwas. That was fantastic. I mean, there was so much there. Anyone who needs an argument for someone in opposition to the use of glyphosate, I think has it now. Makes me think about the fact that it's difficult to argue with science. The same as it's difficult not to vote for science or someone who supports science. On that note, please field any questions.
This is a tough one. This is one of those things where we had this GMO issue a long time ago, back in the early 2000s. And what I discovered was it was just so much easier to get the general public afraid than it is to get them unafraid. much easier. And so that's as a scientist, and then the only arguments you have are arguments that you've had in discussions you've had with fellow scientists, and it's or people who are involved with the tools. And those arguments just fall on deaf ears when it comes to the general public. So this is bad news for.
It's kind of a broader comment on our times, you know?
Yeah, yeah.
Are there any questions in the chat box? I'm trying to see there.
There are a few on the Q&A section. I don't know if Eric, you want to pull that up.
Oh.
I can't access
just really quick, I want to tell everyone that I'm going to be launching the fourth poll. However, the first question is a multiple choice, meaning that you are to choose 3 of the options, and correct me if I'm wrong, Scott, but I'll launch it right now and you guys are going to be able to see it. So the first one, there's 3 of them are correct. Choose 3 of them, and then the second one is just one or the other.
Thanks, Hugo. If we can't access the questions, do you mind dictating them, perhaps? Not sure.
Sure, from the Q&A.
Yeah.
There is.
There's one question I see here about the adjuvants in Roundup. Has there been any research on that? And the answer is yes. So that is becoming more and more of a focal point. And so I mentioned it in my talk. I mentioned, what are the formulations that people are being exposed to? And I talked about there are different formulations that are used in North America compared to, for example, Europe. And I mentioned those POAE surfactants, the tallow-based, surfactants. And so, so yeah, that's a question, right? And what people are exposed to when they self-report, they don't know what they're exposed to. They'll just say Roundup is Roundup is Roundup, and they don't know if they're getting exposed to what surfactants might be mixed in with those particular formulations. So yes, that's a very, very complicating question. And so my suspicion is that I don't know. I don't think, we all know what the answer to that whole problem is that you need to follow the label, right? And you need to wear the personal protective equipment that's on the label. And there's not any reasonable logic that you should fear the cancer scare. It's so that's definitely one additional problem. People were asking, people were asking about field bindweed. One of the things you could do, so like, for example, using shielded, if you're going to spray glyphosate after bud break, that's the only chance, right? That the only possibility, and it's super low for the reasons I gave in my talk, to spray, somebody had said using shields, right, to prevent any kind of a coverage on the grapevine leaves. So anything green and growing is what will absorb glyphosate. And I don't know of any studies that have shown that glyphosate sprayed on soils that get into soils, that they can be absorbed through the root systems of, for example, a grapevine. I don't know of any of those. And what would happen, of course, is that the grapevine would die if there was, significant amounts of glyphosate to kill a weed population. You would also kill the grapevine. So unless vineyard managers start seeing that kind of problem, which I've never heard of, but again, it's perception. So instead of using Roundup, is there something else you could use? Say field bindweed. Field bindweed is a really tough one. I think you all probably know that, especially in Northern California. That was the #1 weed that we saw. And so something like maybe something like shark, carfentrazone, could maybe be some kind of an alternative. Glufosinate is another one, right? That could be a possibility that you might try. So those are, but there's, you know, I hear also alternatives to glyphosate. This is one thing I think should be pretty important to folks, is that the paraquat solution is not the greatest. That's not a good solution. There's several reasons for that. One of the biggest ones is that it's very, you know, it's extremely toxic, as you all know. So let's not go down that path. There's another one.
All right.
Here's a recommendation for tree of heaven. So the easiest way to get rid of tree of heaven and other tree-like species with secondary growth, the easiest way is to, I'm going to go back to glyphosate. What you do is with tree of heaven is you cut it down as close to the base as you can get, and do this in the late summer, early fall, because what's happening is all the photosynthetates are starting to go back underground. Okay, so cut the tree, cut the stumps, and there'll probably be many of them because tree of heaven suckers like crazy, right? So cut the tree and cut all the suckers that are coming up, and within 1 minute of cutting it, be ready with a bottle of concentrated glyphosate and squirt it on the vascular cambium, which is the outer ring just inside the bark. Concentrated Roundup on that vascular cylinder, the vascular ring, right? Which is, like I said, just inside the bark is where that vascular cylinder is. Spray concentrated Roundup within 1 minute of cutting the tree, and do it in the late summer, early fall, when everything's going back underground. You will probably have to come back the next year and do it again, right? And do it the same way, but you'll get it. You'll get it with that technique.
All right. Thank you very much, Dr. Stenmus. If you don't mind hanging out just to see if anybody has additional questions, you know, please take those in the chat function, and we will move forward. Next up, we have a wonderful presentation for everybody that is going to be put on by, let me grab my notes here. Excuse me, where are my notes?
You have Jake Blim from the Ag Commissioner Office.
Yeah, I'm trying to find, Jake sent me his bio here a moment ago. Jake Blim with the National Napa County Agricultural Commissioner's Office. Jake is an inspector, pest detection, pest eradication, and in pesticide use space. He has worked for private enterprise, businesses focused on insect rearing. He's also spent time in the nonprofit sector, working with the Rodale Institute, and he's done some overseas projects, spending time in Bangladesh, Guatemala, Vietnam, Malawi, South Sudan, and Rwanda. Jake received his bachelor's degree in agricultural business and economics at Colorado State University, and his MBA at the California Lutheran University, and he holds a certificate in international management from the Thunderbirds Global School of Business. Jake's going to be talking about pesticide labels today, practical information that we all need to pay attention to. Jake, thank you very much for being here.
Good morning. Good morning. So we are going to talk about labels today. All right, can you guys see that okay?
Yes.
All right, so we need to read our disclaimer here. Reference to commercial products or trade names is made with the understanding that no discrimination is intended for those not mentioned, and no endorsement by Napa County is implied for those mentioned. The following presentation is offered for informational purposes only and is not intended for to be the be-all, end-all. So labels, let's talk about terms. We're all familiar with labels. We look at them almost every day, I would guess. Everything from a nutrition label to a technology label that that might be something that we're diving into. So labels with pesticides are quite important, and they accomplish a number of things for us. Of course, reading a label is your first step to choosing the right product for your needs, keeping you, your children, workers safe, saving money, and also helping the environment by using the right product. Labels tell you how to use the product safely and effectively, how to store the product safely, first aid instructions, as well as phone numbers to call for help or more information on the product. So definition of terms, by the end of this section, we'll be able to define what a pest is, define what a pesticide is, and identify products as pesticides or not pesticides. So what is a pest? Pest defined by the Food and Agriculture Code section 12754.5 as any of the following: is or is liable to become dangerous or detrimental to the agricultural or non-agricultural environment of the state. Any insect, which is our typical primary target, but also predatory animals, rodents as well, nematodes or weeds. Any form of terrestrial, aquatic, or aerial plant or animal, virus, fungus, bacteria, or other microorganism, except these that might be on a human or animal. Anything that the director by regulation declares to be a pest. So then what is a pesticide? Food and Agricultural Code 12753 defines a pesticide as any of the following: a substance or mixture of substances which is intended to be used for defoliating plants, regulating plant growth, for preventing or destroying, repelling, or mitigating any pest which may infest or be detrimental to vegetation, man, animals, or households, or be present in any agricultural or non-agricultural environment whatsoever. Also, any spray adjuvant. And we, most of us understand that the state of California has a more extensive list of what a pesticide actually is than most of the rest of the country. Lucky for us. So I'm going to go through a series of products that are a little bit different from each other, and we will determine whether these are in fact pesticides or not. So product number one is sprayed on apples to promote uniform ripening, to facilitate efficient harvesting. Is this a pesticide? Yes, it actually is a pesticide. Plant regulators, as well as defoliants and desiccants, are specifically included in the definition of a pesticide in the state of California. Product two is an athlete's foot remedy that kills or slows the growth of fungus on living humans. Is that a pesticide? One might think it would be, but no. The product is not a pesticide. Products intended and labeled for use only for the control of fungi, bacteria, viruses, or other microorganisms in or on living humans or animals are not considered pesticides. Those are under the auspices of the FDA. Product three, and this is a tricky one. It's added to horse feed. It passes through the digestive tract and prevents fly eggs from hatching in horse's manure. This is known as a feed-through larvicide. This is a pesticide. It's not it does not, it is not an animal drug because it's fed through the animal, but does not actually impact anything on or inside the horse, just simply a delivery system for that larvicide. If the product is fed to an animal to treat a parasite afflicting that animal, then of course it would be an animal drug, and again, regulated by our friends at the FDA. Product 4, a liquid is added to water lines that supply the rinse water for a patient in a dentist chair. The label says it prevents a buildup of biofilm. Pesticide or no? Yes, it is. This product, slime and algae and biofilms are considered organisms that grow in water, and controlling them is in fact a pesticidal function. Product 5, household bleach. Label says it cleans and deodorizes, but we know that bleach does affect certain microorganisms, right? No, this is not a pesticide. Clean and deodorized are not pesticidal claims. However, if the bleach product label claimed to sanitize, disinfect, or kill germs, then it must be registered as a pesticide. Again, the same material, depending on the intention of the manufacturer and or the marketer, determines whether or not that is a pesticide. Product 6, a chemical additive that reduces or hinders the formation of foam in a pesticide mixture. Very common in our corner of the world. In fact, yes, these are considered pesticides when they improve the pesticide's performance or the physical properties of the spray mixture. Again, in California, and this may not be the case in other states. Product 7, ethyl alcohol sanitizer. A lot of this has been used in the last 7 or 8 months, killing 99.99% of germs. No, this isn't used to kill this is used to kill microbes in or on a person. This is the determining issue. Then it is not a pesticide. It is instead regulated again by FDA. So even though it's something that would impact the microbes, and we hope it does, because it's used on or in a human or an animal, it's not considered a pesticide. This little illustration just shows that the applications should be in the correct environment, and not everybody will want those applications, but we hope that they're effective if they are used. All right, in this section, we want to be able to determine the difference between registration types. We will define a master label and then supplemental labeling. So the U.S. Environmental Protection Agency has 3 main pesticide registrations. Section 3, this product has a standard registration. This covers a very large swath of the products that we use in agriculture and non-agricultural systems. Section 25B are minimal risk products that have been exempted from registration. Many of these are used in organic systems, and some of them are quite common, food commodities, animal feed items, edible fats and oils. One of the last, I think the last weed control presentation talked about cinnamon oil and clove oil. Those would be considered minimal risk products. Section 24C, a pesticide that has been registered a special local need can be established to control something that is, for example, Botrytis cinerea, gray mold on blueberries for export to some countries, including China, India, Japan, UK, and Vietnam. Section 18, a product that's been given an emergency exemption. This example, CA20-03, allows Intrepid to be used on rice to control armyworm. Many years ago, back in the day when glassy-winged sharpshooter just came into Southern California, they were trying to figure out what to use on it, and I think they got a Section 18 for kaolin because they had determined that would be a inhibitor of GWIS on feeding. So they were able to get that through fairly quickly. If you're going to put a new pest on a label, it can take many months or longer to get that actually on the label. So a product requires registration in California if US EPA, Office of Pesticide Programs, requires registration of the product, excluding plant incorporated protectants like BT cotton, for example. And the product is sold, distributed, or used in California. California law requires registration of the product even if US EPA does not. For example, as we mentioned earlier, spray adjuvants, structural pest control devices, other FIFRA 25B exempt products. So even though federally it wouldn't require, California has more stringent pesticide laws, so it would capture these other types of products. So a master label defined by DPR is a pesticide label bearing most or all the EPA accepted uses for that product. However, the company does not intend to market that label for sale and use in California. A supplemental label, on the other hand, that's a term used to describe the partial labels that are distributed separately from the container label. We've seen these many times. They may not be attached the same way as the main label, but they are distributed by either the registrant or the licensed distributor in the state. All right, so what does our label actually include here? There are a number of items that we can learn and benefit from on the product label. A restricted use pesticide has the potential to cause unreasonable adverse effects to the environment and injury to applicators or bystanders without additional restrictions. The restricted use classification restricts a product or its uses to use by a certified applicator or someone under a certified applicator's direct supervision. This product is restricted use due to toxicity to fish and other aquatic organisms. For retail sale and use only by certified applicators or persons under their direct supervision, and only for those covered by the certified applicator's certification. The ingredient statement is another important section of any label. It identifies the name and percentage by weight of each active ingredient, as well as the percentage by weight, but not typically the names of other inert ingredients. You can see here on this label, the AI is 22.6%, and other ingredients make up the balance, 77.4%. And they don't, they're not required to list the actual inert ingredients. This slide just shows that others have different viewpoints of what a pest might be. So we need to be cognizant of that. The signal word is also quite important. The acute toxicity studies examine a product's toxicity as it relates to 6 different types of exposures. Acute oral, acute dermal, acute inhalation, primary eye irritation, primary skin irritation, and dermal sensitization. So with the signal word of danger or danger poison, this is our class 1 or the most highly toxic pesticides. They can be corrosive or irritant properties. The toxicity, can be lethal from a few drops to a teaspoon. So we want to be quite careful with these products. Oops. The second category is a warning label. That's a class 2. It's moderately toxic. Lethal doses are typically a teaspoon to an ounce of product. Signal word caution is a class 3, slightly toxic. 1 ounce to 1 pint or pound would be considered the lethal dose. And then the caution or none, class 4, slight hazard over a pint or a pound of product to be lethal. The next area of the label is directions for use. And again, we want to make sure that we're using these products properly to ensure their effectiveness, their safety, and minimizing any environmental hazards or otherwise. So the directions for use provide instructions to the user on how to use a product. It identifies the pests or pests to be controlled, application sites, application rates, and any required application equipment. So you'll see the basic directions for use. This is typically the very minimal violation under federal law to use this product in a manner inconsistent with its labeling. The agricultural requirements are typically more detailed. You've got worker protection standards in 40 CFR Part 170. The standard contains requirements for the protection of agricultural workers on farms, forest nurseries, and greenhouses, and other handlers of agricultural pesticides. It contains requirements for training, decontamination, notification, and emergency assistance. It also contains specific instructions and exemptions pertaining from the statements on this label about personal protective equipment, the PPE, and the restricted entry interval, which is important as well. Requirements in this box only apply to those uses of this product that are covered by the worker protection standard. PPE, again, required for early entry to treated areas that is permitted under the worker protection standard, and that involves contact with anything that has been treated, such as plants, soil, water. That PPE is coveralls, chemical resistant gloves, and so forth. Storage and disposal instructions, another important piece of information. storage and disposal instructions provide the instructions for storing the pesticide product and for disposing of any unused pesticide or pesticide container. Many of you participated in our container recycling event just a few days ago. We appreciate that, and it's one of the ways that we try to facilitate that that disposal of pesticide containers here in the county. So you can see by this, storage is important. Store in a cool place, protect from excessive heat. Store a product in original container and only away from water, food, and feed. Again, this is to keep anybody safe that might come in contact with that, and also that the product while in storage is going to be safe. Pesticide disposal: do not contaminate water, food, or feed by disposal. Improper disposal of excess pesticide spray, rinsate is a violation of federal law. Precautionary statements, another important piece for us. Precautionary statements are designed to provide the pesticide user with information regarding toxicity, irritation, and sensitization hazards associated with the use of a pesticide, as well as treatment instructions and information to reduce exposure potential. So precautionary statements typically cover 4 sections: hazard to humans and domestic animals, first aid statement, environmental hazard statement, and physical or chemical hazard statement. So hazard to humans or domestic animals. Where a hazard exists to humans or domestic animals, precautionary statements that describe the particular hazard, the route of exposure, and the precautions to be taken must appear on that label. And again, this shows the basics of that precautionary statement. May be fatal if swallowed, causes substantial but temporary eye injury, do not get into eyes, on skin, or clothing. Wear protective eyewear, causes skin irritation, harmful if inhaled or absorbed through the skin. Avoid breathing the vapor or spray mist. Wash thoroughly with soap and water after handling or before eating, drinking, or using tobacco. Remove contaminating clothing and wash clothing before reuse. And the PPE section is also critical, describes the amount of PPE and specifically what should be used. We had an inspection not long ago where the guy at Mix and Load was using gloves, but he wasn't actually using chemical resistant gloves. He's using a very thin nitrile glove. So we had to make sure that he had the correct gloves in his truck and went to get those so he could continue on being in compliance. Discarding clothing or other absorbent materials that have been drenched or heavily contaminated. This is an important item as well. You might think that you can get that contaminant out with washing it, but sometimes that's just not possible. Better to be safe and discard that. First aid statement. This section of the label provides for information to the pesticide user concerning appropriate first aid for the various routes of exposure associated with accidental exposure. If the first aid statement appears on the back panel, then there must be a statement on the front panel indicating that first aid information can be found there. Again, this is to help people get the information as quickly as possible should there be an accidental exposure. And this is so important, depending on the chemical, whether you induce vomiting or do not induce vomiting depends on the chemical nature. So it's very important to understand the specific first aid requirements of each material that we're using. Almost always immediately can call poison control center or doctor. This particular label says do not induce vomiting unless told to by poison control center or doctor. Do not give any liquid to the person. Do not give anything to an unconscious person. It seems pretty straightforward. The eyes is a very common way of being exposed. To rinse eyes, we typically hold them open, rinse slowly and gently with water for 15 or 20 minutes. That's why it's so important to have that close by if you should get product in your eye. If on skin or clothing, take off contaminated clothing, rinse skin immediately with plenty of water for 15 to 20 minutes. Again, call poison control center or doctor for advice. If inhaled, follow the procedure as well. Environmental hazard statement. Where a hazard exists to non-target organisms, precautionary statements that identify the hazards and necessary precautions must appear on the label. This is interesting. It shows the little fish here going belly up. We certainly want to avoid that if possible. The environmental hazards on this label state that this product is extremely toxic to fish and aquatic organisms and is toxic to wildlife. Do not apply directly to water or to areas where surface water is present or to intertidal areas below or below the mean high water mark. Do not apply when weather conditions favor drift from areas treated. Do not contaminate water when cleaning the equipment or disposing of equipment, wash waters. And many of you have participated in fish-friendly farming here in the county. I was out with one of my colleagues several months ago, and the mix and load site was not terribly far from the Napa River. Fortunately, the pitch was away from the river, but again, rinsing off equipment is really important that we be extremely careful about where that runoff might go in that activity. Physical or chemical hazard statement. Hazards such as flammability, explosive potential, or electric insulator breakdown, as well as the various precautions to be taken, must be identified as applicable. Again, you can use the product safely in the vineyard or the orchard, but if it's not stored properly, you can still have a problem. This one, again, showing a violation of federal law to use it in a manner inconsistent with the labeling. It's important that you or your employees, read the label completely before using the product to make sure that we understand what these hazards are and how to mitigate them. So in this section, this is kind of a bonus section. We want to understand label information that might trigger, say, additional personal protective equipment, a site posting, or other actions that aren't listed on the label. Again, for non-ag use, handling or warning or danger, the decontamination site for employees handling pesticides for other uses other than commercial or research production of an agricultural plant commodity shall be within 100 feet of the mixing and loading site when they are handling pesticides with the signal word danger or warning on the label. Again, ag use decontamination facilities are required for employees. So protective eyewear required. This is CCR section 6734, handler decontamination facilities. 1 pint of water for emergency eye flushing should be immediately available to each employee and carried by that handler or on the vehicle or aircraft the handler is using, if the pesticide product labeling requires protective eyewear. When the handler is mixing or loading a pesticide, then only the requirements in the section 6 apply. And again, having that bottle right with the handler should they be some distance from the mixed load site, they're going to need that water to get their eyes cleared as best they can to get back to a steady, clean stream that they can use to clean out the eyes. At the mixed load site, you want to have immediate access to at least one system capable of delivering gently running water at a rate of 0.4 gallons per minute for at least 15 minutes, which is at least 6 gallons of water in containers suitable for providing a gentle eye flush for about 15 minutes emergency. If the product labeling, again, requires protective eyewear or a closed mixing system is used. These are some of the different eye wash systems that are available. Field postings. The REI, in this case, greater than 48 hours. The operator of a property shall assure that signs are posted around treated fields in the following circumstances. Any application that results in a restricted entry interval greater than 48 hours as specified on the product label, unless access to the treated field is controlled in a manner that assures no employee other than handlers making the application will enter, work in, remain in, or walk within a quarter mile during the application and the restricted entry interval. So again, that's a fairly high bar to reach. So having these posted signs is critical for REIs greater than 48 hours. And again, the ag use requirements are covered here. Again, in the worker protection standard, which is the 40 CFR Part 170. Applying pesticide toxic to bees, section 6654. Notification to beekeepers. This is not as critical in Napa County as some other counties that are have crops that are more dependent on pollinators. Down in the San Joaquin Valley with areas that have large incidence of almonds and other crops that citrus that require pollination. Then there's a lot more bees around. But that being said, we do have more bees here in the county than a few years ago. So each person intending to apply a pesticide toxic to bees to a blossoming plant shall, prior to the application, inquire of the commissioner or of a notification service designated by the commissioner whether any beekeeper with apiaries is within 1 mile of the application site, has requested notice of such application. We have this Beware site. Some of you are familiar with that. If you're not, please give us a call at the commissioner's office and we can help you through that. It's important to to check this pretty regularly, because bees do move in and out of different locations, sometimes quite quickly. So it is important to check to make sure that there aren't bees in that area if the beekeeper has requested notification. You would want to do this at least 48 hours in advance of the application, of the time and place of the applications to be made, the crop, the acreage to be treated, the method of application, the identity and dosage rate of the application to be applied, and how the person performing pest control may be contacted by the beekeeper. Sometimes, it's just a text or a phone call. So that's important to know that. This time may be increased or decreased by the commissioner or by an agreement of both the beekeeper and the person performing the pest control work. This section applies statewide. There is from March 15th to May 15th in citrus bee protection areas, there are conflicts between provisions of this section and those of 6656. That section will prevail. Again, we don't have commercial citrus here in the county, but many do. And this is just the end of it. We have different insects that require or prefer different things. They're not all bad. Some provide us with a good lot of services that we'd rather not do ourselves. And that's all I have for today. I can take some questions if any of you have something that you need clarification on.
Thank you very much, Jake. That was great. Do you see any questions in the chat box there?
Let's see. I don't see him here.
Maybe if you stop sharing the screen, there are, I believe, at least 2 questions. One on the Q&A and 2 on the chat.
Thank you, the audience, for bearing with us as we navigate this. This is our first remote experience, and it's gone pretty darn well, if I do say so. And thanks again to Ugo for facilitating it all. I'm going to also take a moment to thank our sponsor, Sunridge Grapevine Nurseries again. Reach out to Julian Clymer. He's the guy up here selling the vines. He's a really good dude, and you'll be happy with the plants. And also, I'm going to throw a plug for our next meeting, November 19th, a week from today. We're talking about climate change. Coping with climate change is the name of the training. We have 5 fantastic speakers. First, Dr. Elizabeth Wolkovich from the University of British Columbia, who's going to be talking about the influence of climate change on grapevine phenology. David Johnson from New Mexico State University, who's built a bioreactor and is producing compost to help mitigate climate change. Annette Reinke, Geisenheim University in Germany, who's going to be talking about the effects of global warming on insect pests of grapevines, and Dr. Mike Treby, who's with Agriculture Victoria, who's going to be talking about rising CO2 levels and how grapevines may function as a result of rising CO2. And then we're going to have Jen Borgen with the Ag Commissioner's Office talking about common inspection issues and how to fix them.
Okay, I do see a couple of questions on here. One is, are we aware of any Beware website improvements in development? When it was first launched, it was very cumbersome to search for your farm in multiple locations. I've only become aware of it in the last year and a half. I don't know if it was more difficult before then. Most people that have used it have said that they're able to get onto it fairly easily. If you haven't checked into it recently, take a look at it again. If you've got any difficulties or questions with that, yeah, give us a call at the office with specific items that might be of concern.
Hey, Eric, this is John Coolidge with the Ag Commissioner. Just to follow up on that question, we are kind of constantly in contact with the developer because we understand some of those features are a little difficult. So we know there's some ways it could be better. So we'll continue to work with them so it can be most useful for for the growers. So we're aware of that. And there's another question, I guess.
Is a decontamination set required for winery employees using SO2 for barrel fumigation?
So I would assume they would, the thing about SO2, it's kind of funny, we regulate it from the standpoint of purchasers, purchasing the SO2 gas, but it is kind of a Cal OSHA thing. It's sort of a crossover between, the Ag Commissioner rules and more Cal OSHA rules. So I would probably refer to the Cal OSHA rule on that, but I would assume that there should be some kind of decontamination for SO2 users. So we're covered by Title III, California Code of Regulations, and the OSHA stuff is Title 8. So there may be something in Title 8. But if the person that asked that question needs more clarification, they could always send us an email and we could do some further research.
I think that's it. Anything else that that comes to mind?
I don't see any more questions. Thank you very much, Jake and John, and to everybody in attendance. We appreciate your support of the Farm Bureau, and I hope this training was beneficial for you. I see a number of questions in the chat box related to issues experienced with the survey, the polls surveys. So if you didn't feel like you were able to complete things effectively, please email Ugo or myself. Actually, Ugo, since you don't have my email address, I don't want to give it out. But we'll get those questions answered, and we'll make sure that everybody is fully accredited for the trainings today. No question. DPR is pretty willing to work with situations like this because we're using kind of a novel, technology here to do something that was handled differently in the past. On that note, anything to add, Ugo, or?
No, I was just going to add on that yeah, if I also plan to follow up with everyone regarding the polls, and there is the last one live right now, so if everybody can answer that one, and all regarding the credits, I plan to follow up with everybody on an email. So
yeah, and then additionally, we do have I put this in the chat, but we have 5 CCA credits approved today too. So if you're in need of CCA credits, please take advantage of this meeting to fulfill those. And if anybody has questions about the CCA credits, they can follow up as well, categorically or whatever. All right, let's put a cork in it. Thank you again. Goodbye. I guess we can keep the chat up in case there are any.
yeah,
any final
I
questions.
plan to leave the poll on for a little bit, and I know that there's people that say that they can't see the poll. I have everybody who I have 83 people answered it, so I'm not exactly sure what could trigger not seeing the poll for somebody in particular, but again, we can follow up on that offline.